you know, optimize.
Yeah, no, I mean, you need to
And
start something.
you can optimize it. They're not uniform. Like what I see, everybody knows that they need to get organized, and I see that working on
But I think, from the discussion yesterday, it seems that a lot of the decision making is really based on internal company and kind of strategy and risk assessment and whatnot. So your job then becomes difficult, right? Because, well, unless you focus on, okay, give us your risk assessment and then we figure
if this is the minimum one MPA or if we call this one good enough. We know this. We know GDP. You know. So I think it should be a good discussion today as well.
can I ask you then a very naive question because I'm fairly ignorant in the biomarker arena. So you get biomarker doesn't mean because it's not going to be a surrogate endpoint, right? You're just going to tell you use this and the FDA is going to have a re-review every time you submit something with this package.
Maybe
Got it, yeah. So have you totally given up on something? Monday.
So you need to so you use it is a justified characteristic of the patient that you can use to either enroll or whatever. But this is still far from the clinical qualification. I mean, in terms of having a surrogate endpoint, this is not to determine this biomarker could be as opposed to this.
It's one step before that.
Right.
It's one step before. Yeah.
And then from there, the theory
Exactly.
I don't know how many surrogate endpoints.
Yeah, you capture that problem. Yeah.
Understood. Good. So where are you located? Enoch. Fantastic. Making connections, I see there actually.
Yeah, I knew these folks. Are we lucky or what? We got Jeff on board to running our extra brain power for our scientists, right? A lot of trainings we're putting together for the
Internal training and external.
you provide certificate.
How about trophies?
Okay, you sure you're not the marketing?
the speakers, and when I'm a speaker, we always end up like at that restaurant.
Yes.
And there's so many choices,
There are.
but it's just wonderful.
Yes, yes. They're very consistent, high quality all the time. I love doing it.
If I'm not interrupting, I want to ask you a quick question that I thought if I asked it at the mic, it might be too off topic. But I work a lot in immuno-oncology. So I'm almost like a datapreneur because I'm a data analyst and a journalist. So a lot of times I go to companies like AstraZeneca, BMS, Genentech, to try to kind of translate some of that so they can understand it with their TPP and all these other, documents. And I feel like I came of professional and personal age during the HIV and the AIDS, where we had EuroResist and we had that huge database of resistance. Do you ever see that coming for biomarkers where we're looking at the activity of the biomarker, not like progression-free survival and overall, we're actually looking in a way that a healthcare provider can make a cocktail. Like, I know this drug does this, this is this, this is that. Yeah. But no one wants to hear it though. When you talk about they're all like microsatellite instability, I'm like, how do you know that's the thing? How do you know PF1 and PF1 expression downstream could be where the effect is? It could be a cytokine. It could be something we don't even know about. Yeah.
This is the final thing. Thinking that this is what happens sequentially effects on the patient.
Yeah.
So I think there's a lot of that is really the problem.
Are you going to talk about any of that around the nephro? That's why I didn't want to bring it up because I thought it was over here. It's
Stop
similar.
holding it
but-
by the antenna too. That's another issue. Shush. 1, 2, 2.
Yeah. Yeah.
It's validated from that particular moment. But as you collect more and more data, you have to evaluate whether the quantity is changing, whether something else is meaning itself from this. Because there's so many adjoining effects that you miss only because you're thinking of this one market.
And what do you think? Because you're in pharma, so what do you think of these the evolving frameworks around like getting away from the randomized control trial and just finding ways to get all the data from everybody so that we can make these decisions?
See, as long as we maintain that in medical practice, because there's so much off-label use of our disease, right? Because of the sad situation in the patient.
Yeah.
That is very hard to say. No, I think it's a good thing to get the drug approved faster.
But don't breakthrough designations. Don't they do it based on the endogenous biochemistry and then they sit back and wait and see what's going to happen anyway?
They do, designation comes when there's a big need in the market for AIDS and they think that patients have nothing and they think that this is the most promising thing.
Yeah.
But that doesn't necessarily always focus on physicians, right? So it's not always that they know exactly that this target is going to be so highly relevant.
Yeah.
Good morning, everybody. We're going to go ahead and get started. Good morning, everybody. I'm Greg Daniel. I'm Deputy Director in the Duke Margolis Center for Health Policy. Welcome to the second day of our public event, Scientific and Regulatory Considerations for Analytical Validation of Assays Used in the Qualification of Biomarkers in Biological Matrices. It's a wonderful title. We had a very engaging and very productive discussion yesterday, and we very much appreciate all of the thoughtful comments and feedback throughout the course of the day on the framework and all of the work that has been done over the last year. We look forward to continuing that productive conversation today with where we'll be exploring our second real-world example and applying the framework, and then turn the focus to next steps in terms of refining and disseminating the framework more broadly into the scientific community. Once again, we have a lot of things to cover today, so I'm not going to spend too much time on background, but I will go through a bit of the agenda today. We'll be kicking off our some opening comments from John Michael Sauer at CPATH, who will provide a brief recap of yesterday's conversation and frame up the goals for today. We'll then go directly into session 4, which will offer the case study framework using GLDH liver biomarkers as the real-world example. Session 5, following that, will allow for discussion of the framework from an industry perspective, and panelists will provide their initial thoughts, and then we'll open it up to a broader discussion, Q&A with the group here, and we invite any comments from the online community as well. After session 5, we'll break for lunch, and then at 1:30, we'll reconvene for session 6, which is intended to generate discussion on methods to disseminate this framework to a variety of audiences. And then finally, for session 7, we'll reflect back on the major themes from both days and prioritize next steps in order to finalize and ultimately disseminate this framework. As a reminder from yesterday, for those of you on the webcast, you can ask questions in 2 ways. Use the comments or live chat section on your YouTube. We have staff monitoring that, and we'll bring the questions to the group. Or you can email questions to margolisevents@duke.edu. And again, we'll do our best to incorporate all of the webcast questions and comments throughout the discussion. And again, today we are live tweeting all of the things today with our hefty hashtag, which is hashtag biomarker assay validation. Please look at that, engage your organizations to participate in the online discussion as well. Again, for today, lunch will be on your own. There are local eateries and food trucks nearby, and then we'd be happy to point you in the right direction. So with that, I'll turn things over to John Michael Sauer. Thank you.
Thanks a lot, Greg. Wow, you guys were great yesterday. I really enjoyed the conversation that happened, and I hope today that's exactly what we can get into a little bit deeper. But what I'd like to do is, first of all, remind everybody about the email, where to not only email questions, but also comments on the white paper. So scribe that down. So what I'd like to do is just, recap really what the scope of this meeting is, right? Just to kind of make sure we're going in the right direction. Like I talked about yesterday in the final session, there is this relationship between the development of IVDs and, getting a cleared assay from the FDA, and also the use of biomarkers during an IND and the assays that are required to achieve both those goals. We're somewhere in the middle, right? What we really want to talk about are what are the expectations from an assay validation point for the qualification of biomarkers, right? And so we want to stay in that space if we can. We keep kind of weaving to the side, but that's okay. We'll do that a little bit. But I think really coming out of this meeting, what we need are really to define those expectations. And so what are the core expectations around assay validation for the qualification of any biomarker? I think there are a set of core properties that need to be defined, and we went through that a little bit. I think we need to refine that a little bit more and have further conversation about that. I think also what we need to do is agree upon how that context of use, the biomarker's property, the patient population, actually define additional expectations for these assay validation approaches. And then the whole objective is to then codify this in the white paper. And so we need your input. This white paper is not done. Our goal, as Steve Piccoli nicely laid out, was to get us 90% there, right? To get to do the easy part. You guys have to do the hard part, have to get that last 10% for us so we have all the right things in that white paper. And of course, that we're not overreaching, right? We're not asking for too much. So I think the sessions yesterday went really well. I think we got some great input, some great questions, some great conversations that went on. I took, I don't know, page upon page of note, and I'll share some of those, high-level observations that I saw. I think we did come to agreement that the context of use really drives the assay validation for the qualification of a biomarker. I think also the idea that these core expectations are there. Now, although the approach that's used in the use of biomarkers in an IND are very similar to what we're talking about, I don't think the expectations are as high. I think we heard that yesterday, right? We're not talking about those same level of expectations. So I think that's really important to note. I mean, it was said, through multiple stakeholders, from industry, from FDA. Sure enough, there's expectations, but they're not going to be to the level that we're going to be expecting for biomarker qualification. We also talked about that this is a group effort, right? It's not done in isolation as far as the qualification of a biomarker assay for qualification, or I'm sorry, the validation of a biomarker assay for qualification. It's going to require the clinician, biologists, statisticians, and yeah, of course, the bioanalytical scientists to drive those assays. I think we also agreed upon the fact that we just can't have a checklist at the end of the day where we're going to be able to go through and just check the box and finish these assays. Instead, it's going to be a framework, right? You're going to have to use the science to drive what that assay needs. Finally, and it was interesting counterpoint around how the CLSI guidelines or documents should be used, right? And it's what I got from that conversation is that, the CLSI guidelines are made for in vitro diagnostics. We know that, right? That's what they were designed for. But I think there's clues in there on how we can solve problems and issues that we run into in the validation of biomarker assays. So that's what I got out of that. We can talk a little bit more about that as we move forward. And I think also in the last part, the last conversation we had, or the last section that we had, and the conversation we had there is, I think there's an expectation for these biomarkers when we engage the FDA for qualification that they're regulatory ready. And I think we need to flesh that out more. There's no doubt about it. But it sounds like by the time that we get to the biomarker plan phase, or the qualification plan phase for a given biomarker, we need to have a validated assay. So that's going to take a lot of additional work that I think before we weren't offering the agency when we engaged in assay qualification. And I think there's also the recognition that there are several areas in the white paper that we can still add and enhance, which I think is a good thing. I think we also really need to clarify the fact that for the early exploratory studies for a novel biomarker and trying to understand whether this biomarker is useful or not, so pre-qualification, you don't need to have a fully validated assay, right? And I think that was the whole objective of Table 1, although I think we need to couch it a little bit different. So that's definitely an area in the white paper that we need to modify around. I also like the idea of this process map or this roadmap to qualification and embedding that in the white paper. There are several other points. I couldn't list them all out, but we have them in notes. So I think one of the other subjects that came up several times, and I think this is absolutely key to the white paper, is, are we asking too much? I know bioanalytical scientists, you guys want to have really good assays. You're proud of your assays. Of course you are, because you want to generate data that can be used. But the question is, do we need that much rigor and robustness around these assays for given biomarkers? And so I think we need to be careful that in the white paper, we don't over-design this and ask for too much. There's no doubt that the key elements that are shown below, are needed. But the question is, how far do we dive into that? How robust do we characterize those various aspects? So again, I'd like to make that a part of the conversation today as well as we move forward. So, Greg's already gone through this. We have an exciting agenda here, I would say. I'm really looking forward to the further conversation and driving this forward. But I think what everybody's hoping is that we can anchor at certain points and come to agreement around certain ideas and aspects, because that's really going to drive into that's going to really drive the white paper itself and allow us to complete our task. So with that, I'd like to go ahead and introduce Yuri Albrecht from Pfizer. He's actually the co-director for the PSTC. He's going to talk to us today about the GLDH assay validation and a little bit about the qualification. Yuri?
Yep, thank you very much, John Michael. Hey, good morning. Thank you, everybody, for coming. And we'll spend some time on discussing this example, which it might be a little unique because it allows us actually to go the whole way. Let me just whether I know how to do these things. All right. So we'll have a panel, and the panel discussion after that, and there's going to be Shelley Shoemaker, Julian Lessard, and John Michael Sauer joining me here to discuss the topic. Okay. So now we talk about an example. In this case, it's a glutamate dehydrogenase as a liver-specific biomarker for hepatocellular damage. So one needs to first talk about, what is the unmet need? What are we actually solving? So the unmet need is that the gold standard for hepatocellular damage, the leakage alanine aminotransferase, is not really specific for liver. And I don't know, people don't appreciate that actually that biomarker was discovered as a biomarker of myocardial infarction because it's present in the muscle. And after that was kind of a thought, oh, it's actually good as a biomarker of hepatocellular damage. And that's how we know that marker today, okay? So, but the problem there is that once you have a muscle injury, you get a release of that ALT enzyme, and you are not able to differentiate muscle and liver injury. You're not able to diagnose liver injury on the background of muscle injury, okay, using that enzyme. So the case, why is it needed is, in that table, you see a situation in a subject with hereditary muscle diseases, okay? Those subjects have a high levels of ALT and AST, right? So if you imagine that you got a child with Duchenne dystrophy and then has a fever and the mother gave him acetaminophen and then get the cough drops with acetaminophen and so on. So you can overdose that child without even knowing. You would figure it out when the bilirubin will go up, but that could be too late, and you actually are losing the time for administering N-acetylcysteine as an antidote and so on. So it's kind of a case, kind of for consideration. The other one is a subject with acquired muscle impairments, rhabdomyolysis, myositis, and so on. And those are people a lot. It's pretty prevalent, right? Some drugs cause it. So, and then it's difficult to say, okay, is it liver or is it muscle, right? So on that left side, you see that the correlation between alanine aminotransferase and creatine kinase, which is the muscle-specific biomarker. So it's basically, they correlate together, which indicates in the subject with the acquired impairment, muscle impairments, the ALT is not really useful, okay? So, so the answer to that is the GLDH. And the GLDH, I call it the gift which keeps on giving. Okay, it's a very interesting story. It's been around for quite some time. It's a simple enzyme which it's involved in metabolism of glutamate, and it's been looked at as a biomarker of liver injury for long time. Okay, but what has happened is never really got caught up. Okay, there are assays, and we'll talk about it. But actually, there are now really reports, and nobody really systematically studied that enzyme and why, what is the advantage of that enzyme and how it fits into the paradigm of diagnosing of liver injury or liver disease. Okay. So, if one think about it, if one would qualify or use that enzyme, it's like ALT. Instead of ALT is a cytoplasmic enzyme, this is a mitochondrial enzyme. But the story is the same. The hepatocytes don't feel good, burst open, you get the leakage out, and that's how you can measure it in a serum. So, would actually the alternative biomarker to do like GLDH? The impact of it would be able to facilitate development of all therapies for muscle impairments, right? So you have a drug, you're testing drug in a target population. You don't know how you assess that the drug doesn't cause a liver injury, right? The only thing for onset of liver injury, you got ALT, they already have it. So that'd be one thing. And then the second piece is the public health or the patient care, which I'm talking about, and that's the child with the acetaminophen with muscle injury who get overdosed, and how do we diagnose it, right? So there are two kind of impacts which are quite important and worth of investigating. Okay. So just to demonstrate how the GLDH actually works, and this is the same table, I just added the GLDH data for the subjects. As you can see, the ALT and AST as was before, it's the same. It's increased in the DMD boys, which are the boys with the Duchenne dystrophy. And as you can see in the GLDH, the levels of GLDH are exactly in normal range as normal boys or normal adults. So it's not affected by the muscle injury. The other graph on the right side, it's a time course of a subject with rhabdomyolysis who went under through a hypoxic crisis, which leads to secondary liver injury. And as you can see, throughout the hospitalization, development of the rhabdomyolysis, see the increases of ALT, AST and CK, but the yellow line is the GLDH, and the GLDH is normal until that hypoxic shock and that secondary liver injury, where you can see shooting it up, right? So that's a kind of a clinical, a case which documents that you can detect onset of liver injury on the background of muscle injury in those. And that would be very helpful for these people because those cases get undiagnosed, right? In this case, the rhabdomyolysis and the liver injury resolved itself, but in many cases, knowing that there is a liver injury, then influence the treatment options and improve the care of these patients. So that's kind of about a paint the way what the enzyme can do. And which kind of leads me to a context of use, which we outline for our qualification and the risk and benefits. Because I think people need to look at context of use and risk and benefits as we outline in our evidentiary consideration for biomarker qualification. And basically, we have a decision tree there. And what the context of use basically is saying that the elevated GLDH levels in subject with muscle injury indicates a liver injury and can be used in clinical trial in conjunction with care of the patients. So the risk and benefits is we don't believe that there are really additional risk associated with application of GLDH because if we don't have GLDH, all these cases are undiagnosed, right? And if the GLDH would fail, then at some point we'll get bilirubin. We get the bilirubin today, so we are not adding anything more risky to this, right? So what we are doing, we're actually providing a benefit to the subject that we see the onset of liver injury. So just that you understand the context. So now that brings me to the assay, what we are using. So we were lucky in this project because we didn't need to go to a really assay development optimization and so on. So this is really an assay validation exercise here. We didn't move the assay. So the assay is actually manufactured by Randox, or Roche has the assay as a research purposes only assay. It runs on these wonderful machines, and it costs about $1 and time an assay. So the assay is manufactured under pretty standardized conditions, and it's in terms of mechanics of the assay it's very simple. It's a spectrophotometric assay of enzymatic activity, right? So what the validation is really characterization of that assay in light of the and the impact on that context of use, bring that into alignment, right, and understanding the limitation of it. Okay, so that's where we are. So that brings me to a kind of give you the continuum, and I think this is in my mind, my view, the most important things when we talk about analytical validation. This is in my view, it is the analytical validation continuum, right? So I've got an assay, I've got it thinking that I have a biomarker. What do I do, and how do I do through that I get from the exploratory studies through application, through qualification, application in clinical trials, and potentially, in this case, I can actually walk it the whole way to in vitro diagnostics, right? Because it would actually be benefit for the humanity, right? Have that assay, all these subjects available in every hospital. But but thinking about that, and I'll kind of go over these individual kind of a parts of the validation, and seeing the validation as a sort of a living document. Because in my mind, if I have an assay which is made, we are not changing it. So what I'm doing, I'm characterizing the performance of that assay in light of how I'm using it. So I'm always adding it based on how I'm using it to it, making it more detailed. But it's the same assay, and I'm using it in light of what I know about the assay, right? That's the important piece. I'm using the assay, what I know about the assay, or know how the assay performs, because that's important for the interpretation of the data. So how do you start, right? So you got an idea to develop biomarker for liver injury. So you start with some exploratory studies. So that's how we started. So you validate your assay, and so the goal of it is to understand, get an idea how the biomarker perform, and write a paper about it, right? So I call that basically a validation, which everybody does in academia everywhere for a good science, that I have a reproducible data as I'm using it in a single laboratory with the goal of publishing a paper. And that's what we have done. So you need to characterize all these things, actually all those 7 categories anyway, but it's for one laboratory, it's for the set of subject or samples I'm getting, and that's how I do it. And then I have the paper out of it that we published in 2013. So then you get the understanding how the biomarker behaves and with your assay, and now you just say, okay, now I really think it's going to be useful. So I do some context of use studies, kind of confirming, variety of subject with variety of diseases, muscle, and so on and so forth. So for that, again, I'm running in my laboratory. Again, I'm building on that initial validation which I made for the paper, and then maybe I'm using samples which were frozen longer. So I just need to do stability, that I cover that part for the freeze stability, or samples they were handled differently. So I need to have that. To me, it's more of a thinking guide of looking at where the samples are, where the subjects are, what these things then, what these conditions of use are, so that I am addressing them in my validation, right? And in my validation, I do it the way that because if I know that, say for example, I have samples which are frozen for 2 months, and if I do the 2 months stability, free stability, and it doesn't pass, then I know I cannot use these samples, right? So it's kind of a common sense in my mind. It's just kind of adding to it. So then, in our case, now comes this kind of this green green rectangle, and then we are going to the qualification or application of the biomarker under the IND. Okay. So in our case, we actually went first to get the biomarker into clinical trials under the IND because of its utility for developing therapies for subjects with muscle diseases. Okay, we did that first, and now we're doing the qualification. And that's the wonderful thing, which once you go to that route of using that biomarker as a decision making. Okay, we're not talking about a biomarker which is used for internal decision making of the company, of an exploratory marker. We're talking about marker you're bringing to a clinical study where you're making a decision on discontinuation of therapy. We're bringing the marker which is really characterizing a liver safety of a subject. So that bar for that particular trial under the IND, it's pretty high, and you need to really have that assay validated the way that you add to it this requirements which CLIA asks you for, which is the proficiency testing and which is the method comparison. And the reason for the proficiency testing there is that you have a trial which takes a year, right? So you measure these samples on basis, as the samples come to the laboratory, and you want to make sure that your data in January are the same as if you measure them in March or June, right? So you need to have the proficiency testing and so on. So you are adding to it, and again, it's how you're using it, right? So, in my mind, that bar is higher than the qualification bar, because the qualification bar is saying I've got an assay which supports the context of use, right? And I don't need to run it in CLIA laboratory. I'm having it be good enough that I can interpret the data for that context of use. Okay. Once you go and once you apply, then the sponsor needs to bring it into a validation status that he can make medical decisions on it. Okay. So that's my kind of a view on it, how I'd like to see it. And after that, if you want to go to in vitro diagnostics, that's a totally different can of worms. But I would like to see all this continuum sort of fit together, that you can work and use at least parts of it, even though you may need to do some other studies and address it a little bit differently in different stages. But at least you can capitalize on that experience and expertise you develop and the data throughout the continuum. Okay, does that make sense to everybody? Okay, and you may disagree. I might take it apart during the discussion. Okay, but I think something which would provide us as a guide of thinking, and considering that every situation is different, right? But we need to have a guide of thinking how to move from place A to place B. All right. So then, what I've done, looking at these, progressive validation of biomarkers, and I put together. This is basically a little bit more detailed. Those 7 categories what John Michael showed us, and just put them, together as looking at what do you need for the exploratory, say the paper. A kind of a validation assay for a paper, because it doesn't necessarily mean that for a paper I have a crappy assay. I need to have a good assay for the paper, right? But you can see, I just made these examples. If I'm using samples immediately, or I don't keep them on room temperature, just freeze them immediately or put them in the fridge. I don't need to have a stability at room temperature, right? Because it's not the condition of the use. So that's kind of for illustration. I don't need to have a method comparison or proficiency testing. It's for a paper. So then we move to the biomarker qualification, where will be used more. It will be used in variety conditions. So I need to fill out the blanks in it, but I probably don't really need a proficiency testing on that because it's not going to be that lab who's going to be measuring it, right? So I think the lab which eventually will be measuring it would need to know these things, right? But it's not really necessary for the qualification. And then we'll go to the laboratory developed test. That's the clear standards for applying it for decision making in clinical trials, where you got to have the proficiency testing and so on. And then there's another box with the IVD validation, which I'm not going to go to, but that's the detailed kind of way how you validate the assay. And in my mind, it's all kind of it goes along the lines is, an assay which is done by a single laboratory, trained personnel, to IVD, which is done in all laboratories, and it has to be really foolproof that anybody, can do it routinely, right? So that's why you need to characterize the assay more and more in order to be able to know the limitation that you are able to assure that the assay can be interpreted in every laboratory around the globe. Okay, so that's kind of how I kind of think about it when I think about the level of effort you need to put in to characterize the assay. Okay, so the question here is, we can spend and haggle here all we want, about what actually consists, and this is for illustration purposes from the white paper. If you think about the variety of effort you put in the different stages, right? You can go from 60 samples to IVD 2,160 samples, right? To achieve a precision. In my mind, precision doesn't change. The precision of an assay or how the assay works doesn't change. What change here? How well characterized I have it. I'm not making the assay better. It's not wine. Assay doesn't get better if I do more. Keep wine longer, it's maybe get better. But assay does not. It's the same. What does it do here? It's my SDs will become tighter to a point. And then if I do 2 million samples, it's not going to be better as these anyway, right? So it's about it's about level of characterization of the assay. The assay does not change, and how much I know about it, and I need to use that information in terms of interpretation of the data. Okay? Hopefully it makes sense to everybody. All right. So now, again, this is kind of a flashback to these categories, what we are looking at, and then I have here, 2 slides. I don't want you to read it, but just kind of I took it from a validation report for the GLDH assay and how we address it. The beauty about this is that we can see, we can skin the cat many ways. So there's always other way how one can do it, and in terms of the acceptance criteria. And I think we need to get to a point that we'll get some guide and thinking about what those could be and how we're going to do it. The important piece I wanted to show here is the feedback which we got from the agency, which I think was fantastic. Okay, because in terms of precision, we got like, oh, you guys. We had precision, right? But did you really do the precision around your cut points, right? Because we had one low and one high, and the one cut point in the middle, we didn't have a precision on that, right? It's linear, right? But I think it's a great point again, which signifies the confidence in your cut points, right? So that's, I think, was cool and revealing. And then, of course, you can talk about the accuracy and what is the clinical truth, and comparing it to another FDA-cleared assay. How you can compare something to another FDA-cleared assay if there is none, right? But it's the thinking process, right? It's all about the thinking process. Linearity and all this. So addressing all these issues, and we can have a debate, and I think the white paper should give us an idea, how do I go about it, right? Not, in my mind, not really prescriptive, I need 300 samples or I need 3 samples. But no, you have to sufficiently address this that that the variability does not influence the interpretation, right? And it's up to you to figure it out. I mean, it can be calculated, right? Hopefully that makes sense. I don't know. Okay. So, and then, this is the rest of it, and it comes down to the proficiency testing because we're using it and running it in a CLIA certified laboratory and make a decision on it. And I think I had here another one. When we look at the interference, we did not. If you look at icterus, leukemia, and all these things. But the very interesting point is the interference with exogenous substances, right? So is there interference with drugs or something like that? And it might seem kind of overblown, but think about it. ALT is by itself interferes with a lack of vitamin B6. If you don't have vitamin B6, it doesn't work. Okay, and believe it or not, there are other enzymes, and companies are working on them, which are transaminases. And I've seen in my life a inhibition of ALT by off target, because if you have transaminase, it just goes to that active site, and just zero ALT activity after this. So that's a really good point. So the point is that you address it in the qualification that you're working on a specific population, and you got to make that, in my mind, that thinking exercise. Do I have subjects that treated something, and they have, all these things. But it's a good point to think about it, right? So the question is where it should be, and then people should think about it and address it, appropriately. I don't think there is a prescribed thing that would take all, PDR and take every single drug and try to figure out whether that's going to happen, and hamburgers or whatever, I don't know. And so fruits, so it's, like a grapefruit juice and inhibition of cytochrome CYP3A, right? It's known. So but really, it's about thinking. Okay, so I think I'm at the end of my monologue, and I hope you at least enjoy it a little bit. And we'll go to the panel session. So I would ask, the members of my panel if they can come here. And then you can start asking questions. Okay, I've got some questions here. Kind of a guide to the discussion, but so with the panel. So we all know John Michael Sauer here. So we'll get Shelley. Can you introduce yourself?
Is this on?
Is it on?
Yeah, I'll take yours. I'm Shelley Shoemaker from Pfizer, and I've been involved with the GLD qualification from the beginning, and even way back about 10 years ago when we were doing kind of the preclinical evaluation within the PSTC. So kind of been along for the ride.
And Julianne?
I'm Julian Lessard. I work at the FDA in the Office of In Vitro Diagnostics, Division of Chemistry and Toxicology Devices, and I've been involved in the GLDH qualification process as well.
All right. So now you heard the kind of my monologue about it, and where we are going with this. So I put here a couple of questions or thoughts. What people think. It's not limited to this, but it's about all the validation. So about how we put data. Should we be rigid? Should we be really kind of a thinking and about where are we going? Where it's all bringing us, and then the differences between the drug development tools, the LDTs, the applications, and so on, that we understand it and really don't do too much. We do that we support it. Okay, and also about the CLSI guidelines, and its role in where we are. So I'll just leave it up to you. Is there any question, or you want me to shoot these? Oh, please go ahead.
So actually, a very, very nice story. It's probably valuable to convolute that into the paper to give people that linear path. I really recommend that gets done. I guess that's a job for you or me then, John, right? So one of the things that I asked as a question, though, is there's a couple of different components to what you described relative to, I guess, many in the audience. Method comparison studies. So you're talking about an instrument and a kit. Did you do method comparison studies with split specimens back to the manufacturer or to a different laboratory with a different regulatory structure, perhaps a CLIA lab to a CLIA lab, or your single lab to the manufacturer. Maybe you could just dive into that little piece, because that's a little nuanced.
It was a split samples to another CLIA lab.
Great. Did you check that you were on the same lot of materials for
Yes.
calibration? So you actually did a pre-check on calibrations, lots a lot, QCs, so that you were preloading the success rate. That's very, very cool, and that's very, very important. Thank you.
We did that.
So, Judy, a question here. So, I may have missed this, but can you tell me how early in the process did you really assess bias in the system? Because I noticed in your method comparison, you try to compare two methods to try and understand if there is a bias using human samples. But early on, when you're going through this continuum, when did you start thinking? Because you didn't have, you had, I don't know what your reference standard was, I missed it because I had to step out. How did you know, the bias component there? Since that's a key piece in what we're talking about as total allowable error, I think it's a good thing for you to highlight when in the process you understood that parameter.
So our calibration standards were purchased from Randox, I believe, and they are CLIA approved, so they have verification standards, and there was a range on those, and as long as we met that range, we said that the QC had passed. So I'm not sure we addressed bias actually in the way you're talking about it early on.
Right, but do you know what the characteristics of those standards were that were cleared by CLIA? Like whether endogenous material, recombinant?
They were endogenous materials that they had qualified for a number of assays, not just GLDH, that they use in the clinical labs, CLIA labs.
Okay, so that's definitely a better situation than where most of us are when we're thinking about exploratory markers.
Yeah, that's why I'm saying this is a advantage, right? Where you can actually walk through it. I see this. That's why I'm talking about GLDH as a gift which keeps on giving. It's a good example to walk through, because then you can do what exactly what you're saying, and we can kind of see the impact of it and how we can kind of maneuver with it relatively simply.
Right. So I just want to throw that question open for anybody who has experience to come and talk about when do they think it's appropriate to be thinking about bias. Mostly we are focusing on precision, but the important component of bias, when does it really become real in terms of having the right sorts of materials to assess that?
Actually, I think that was a good segment to what I was about to say to Mina, so we can maybe go back and forth.
Yeah.
Look, I think great work, okay? Beautiful work. But I do think this is 99% of what we deal with is beyond this. And let me explain that. You had a system that is fully automated, almost fully automated. You worked with a manufacturer, I know Randox, that they qualified that standard inside out calibrator. So you had that. You had a biomarker, an assay that has been around for 50 years plus. That assay, you can buy any component and you can buy it from 5 different manufacturers and you can actually get the same results. You had a system that is made for CLIA laboratories to give it to operator, put the sample, push a button, and walk away, and the numbers comes out. So that is not the world that 99% of us living in. So kudos to you. It was great. The market needs it. So as we talk about putting this in a white paper, I just want to make sure that we're realistic with what we deal with. Beautiful work, but I'm not sure it's applicable to a lot of things we're doing.
So the answer to that is I think you still have to address all these issues, right? You still have to address and go through precision, accuracy, and all these things. It may be easier because you've got that laboratory equipment, and you really don't have changing changing antibodies or anything like that. I agree with it, but at least is this the most simple system which you can show we've done through this whole process and get to a point that that the assay is has to be validated for the qualification too, right?
Yeah.
Does that make sense what I'm saying?
It does, and I think I just want to encourage the team for one thing. I think what this audience is looking for and what people are looking, calling in, is that you say, it makes sense. You brought a great example. You said, you're going to go ahead and store the samples for 2 months. So it's no-brainer. Go ahead and do the stability for 2 months. I think what this audience is looking for, okay, I know I got to do a 2 months of stability, But is it that I take 3 samples? Do I take a low, mid, high? Is it endogenous? Then do I, put it in a freezer for, 24 hours and then it's tying? I mean, I think what we need in this industry for what we're talking about is some specific. That again, I go back. There are white papers, there are Gene Lee paper, there are other papers that I think I'm hoping in our white paper. And what is missing is some specifics. No, don't people and say you have to do this, but some level of guidance that this is what is the best practices, and hopefully get the approval, the buy-in from FDA to make this into a guidance. So I just leave it as that. Thank you.
That's a great point.
I mean, if I may speak up, that is kind of what the CLSI guidances do. So these are consensus documents between industry, government, academia to kind of give an idea of some of the studies on how to set up the studies to answer some of these questions.
I want to go back again. I don't want the CLSI guideline numbers to be thrown at me. It has been thrown out for several years. I want to be specific. In the CLSI guideline, show me exactly where they talk about sample stability, not calibrator stability. Show me in there, there are aspects. We keep talking about CLSI guideline. Show me exactly how it is applicable to the work that we do. We have heard it for several years. We need a specific. Get an example of the drug development and show it in there.
It's difficult to do because it depends, on the assay. It depends on the technology. It depends on the context of use, the intended use.
Right. I think that's where, talking to the agency early has really been critical. So even for our assay that was, developed as a laboratory-developed test, when we went to the FDA, we got feedback that we had to go back and do the precision and the spike recovery with our medically relevant samples. So even with the test that's supposedly developed to the criteria for laboratory-developed tests, that the back and forth to the FDA was really critical, and I think you have to be flexible and open to that going forward.
Well, I think one thing that we need to be careful about too, and Ash, we talked about this, right? How prescriptive do we want to be in this white paper? If you look at the guidance, even for bioanalytical, it's not super prescriptive. There's room to maneuver, right? There's no doubt. It's a solid guidance, but there's room to maneuver.
I am not advocating to tie everybody's hand. I've actually been an advocate saying that, do not tie my hand because I got to use my brain, I got to use my science to deliver good quality validation to support whatever that I need to do. At the same time, we got to provide some general boundaries, right? That this is what the industry standard is, that's what we're doing. Get the FDA buy-in that this is the minimum core. That's what is needed, okay? Rather than throwing CLSA, I can go in, if you leave it alone, for me, I continue in doing your good validation work, right? But for the new generation that comes in, do we want them to go reinvent the wheel every single time, having the same conversation we have right now 20 years from now? Or do we want to go at an anchor at some point based on the experience that we have gained and then for them to build up on it? So again, I'm just getting a little bit, the philosophical discussion over and over. We got to anchor at some point something in stake in the ground and move forward from there. If it's CLSI, if it's Gene Lee, if it's somewhere in between, we got to do it.
So do we use our examples to do that? And that's what we've been trying to do because we've been building more and more examples into the white paper. And I think those are the learning points, right? So we need to find more real-world examples.
And what I want are examples. We all put our pretty picture of what worked. What I'd like to see, and actually I would encourage FDA, put examples of things didn't work. Because we can also learn when a validation went wrong, when something was submitted and it was not approved. Why was it not approved? Because the lab could not reproduce it? Or is it that there was fundamental issues with that? So to us is that, as some of these meetings we have, I think we should have a 2-day meeting of examples of everything that did not work and FDA rejected. It would be so valuable.
Well, the interesting part is thinking about the qualifications that the PSTC has been involved in. The FDA has never said, nope, this is the wrong assay, you can't use it. Instead, it's been more around, we need additional characterization around X, Y, and Z. And so we've never gotten to the point where it's been said, sorry, your assay's not going to work for this biomarker.
Okay, so over there.
Thank you. John Allinson, LGC. So maybe address that point in part, but also a question regarding quality control based on your comments, which were very, very good. First of all, you said it was a colorimetric assay, but clearly the assay outline is measuring enzyme activity. And many of these analyzers, and the typical gold standard for that is to use zero-order kinetics rather than a fixed incubation. I just wondered what type of method that was, and the reason for the question is that if you're using zero-order kinetics, the validation exercise for things like Prozone is quite different to the way that you would do it in a standard immunoassay.
Okay, I have to plead the Fifth Amendment.
Okay, that's fine. And I guess to Afshin's, yeah.
Shelley, do you know?
I know it is a kinetic assay. It's all done on the instrument. I know they did look at the substrate depletion and the effect that when they were doing the validation.
Yeah. Okay. So then the other one is things like you can also get cofactor and coenzyme restrictions simply because of very high values. But to Afshan's point as well, you showed it was a fully automated clinical system, but these assays start out as microplate or test tube assays. They don't go immediately on fully automated systems. And again, I've been around long enough to be able to say that that's how we did ALT when it first came out. It wasn't on an automated clinical platform. Right. As to the QC, you mentioned about getting the QCs at the right level. I'd just be interested in your comments because I find very difficult in the bioanalytical world using standard PK quality control in terms of either 4-6x batch to batch, or we don't have enough QCs on there, they're not at the right concentration, which goes to your cutoff level. But also, typically looking at whole studies start to finish where we're using the same quality control material to try and assess true assay performance, because it's very interesting looking at the different levels of validation that you do at different stages. I think one of our biggest challenges is actually getting our validation data to mimic what will happen in the sample analysis stages. A lot of companies put their best teams on validations, they get great data, throw it over the wall to the production team, and it all goes belly up. And so your validation exercise typically may not predict how the assay will work in a routine environment. And there's ways that we can improve that, multi-operator, multi-platform, all of that. But I just wondered about, the idea of accepting critically batch to batch based on the criteria that is used in PK I don't find applies in the clinical world at all. It doesn't add up statistically or clinically. So I just wondered what comments were in terms of how you think we can actually give some leeway to people to look at their quality control in a truly analytical quality control manner, which wouldn't necessarily restrict accepting batch to batch as you go along, but perhaps retrospectively looking at the data overall to show that we've not inappropriately either accepted or rejected data.
Those are all the good points. Yeah, please.
Is that John? Hi.
Hi, Jean. Good to see you.
Yeah, I understand what you say very well. And actually, in practice, I make up QCs using the recombinant. And I use those QC as a pass or fail to accept the run. But in addition, I have so-called sample control. Those are truly endogenous sample pool that I collect and pool and subdivide it into thousands of aliquots. And I use that to learn, okay? And so along the span, I still remember one assay that we had like a 3-year program, and I used them. And you can see the chart, like a Westcott chart, that there are, lot differences. But it is, you can clearly see the bias is due to lot differences, number one. Number two is that I also use that to look at stability trend. So, there's a lot of wealth of knowledge you acquire from the endogenous sample. So I still say that for stability, you have to use endogenous sample.
Yeah, that's exactly how we did it.
Yeah.
You describe it with the pools.
And I do exactly the same and have done all of my life. So it's using sample QC is important. I guess my question was directed as, if we're putting a white paper together to give guidance to the bioanalytical community, how does that sort of thing translate into recommendations for accepting the data of the study as opposed to, you know, the typical way that laboratories run when they're doing PK work, which I don't think applies in the same way. We don't get the right information from that.
Yeah, I mean, in this case, it's even right. The assays are on a routine basis because you monitor safety of subjects, right? So it's not that you have a study and you finish the study, and so it's basically continuously. So that's why the proficiency testing and using these pools and so on, just kind of making sure that you've got the quality assured over a long time, and even your 2 samples you measure on Wednesday not gonna be drifting, you know, over time. So. Yeah, it's what the the people with clinical labs are deal with when they have the laboratory developed test in hospitals, right?
Yeah.
That's the message I think we need to get out to the community that have not done that type of thing before.
Right, and I think we have the benefit of doing this assay in a CLIA-approved test. So they have all those procedures in place already, but we still need to put in the white paper, at least make it clear in the white paper that they do need to have QC samples, even if they aren't available. You need to create them and monitor them over time so you understand the trends. Because I think that happened with safety. In some cases, we didn't have the QC that we needed over 5 years. So
yeah, but I think we need to really clearly distinguish the thing, because I don't want to have everybody who goes with a qualification, and I don't think in my mind it's the goal to go through qualification like it would be a LDT for safety and being applied for that at that stage. I think people then, when they would apply, that they have to figure that out, and that each individual lab has to figure it out how to do the proficiency testing and do all these things. They produce a reasonable data, but I think for the qualification, I don't think we need that.
But we have to be careful, because I think what we've learned is these qualifications take a heck of a lot longer than we plan. Right. I mean, I think the original estimates for the kidney safety program, it's going to be done in 2 years. It's going to enroll quick. You know, we'll be okay.
I know
it's taking a lot longer, right, Steve?
So I'm switching back and forth. It's okay.
Yep. Okay. So I'll quickly go. I wanted to come to your second point there. First of all, I like the presentation a lot. It's a perfect case study of everything going well. Right, and I like the analytical.
Yeah, we made it up.
The analytical continuum, the concept we've talked about, but you laid it out very nicely as well. But I'm wondering if we need sort of a parallel analytical validation continuum for the 90 to 95% of the biomarker assays that are implemented in the clinical trials. So that's your second point there. How can we integrate this to biomarker implementation? And I think maybe we want to clarify that a bit by what we mean by biomarker implementation in clinical trials. Are we talking about the quote-unquote 90%, which include exploratory, target engagement, proof of concept biomarkers that you never ever have intention to take them to predict something other than internal decision making? So this is where it's getting confusing a bit in the audience, because I think there is a premise that these criteria, or what we are discussing, the validation requirements, would then translate down the road into the biomarker implementation in clinical trials as well. This is where I think you're hearing slightly differing opinions about what is appropriate. I think Mina's comment about the bias, reference interval, you never would have a cut point to begin with when you actually are trying to quantitate a biomarker, so-called relative accuracy still, but you want to use it for PK/PD modeling, QSP modeling. So there's a variety of uses for these biomarkers. So it goes back to context of use, but I think it requires some clarification.
I mean, I would go to Chris's comment from yesterday, and Chris, jump into whether I'm misrepresenting what you said. This, to me, is, and I think I'm on the same island as you are in this. If you use it if you have a biomarker and use it for a internal decision making, you're not really about dosing. You're not really influencing patient safety. Do apply it as you wish. What we are talking about here, it's a biomarker which you make decisions on dosing, decisions on inclusion, exclusion, and so on. So, Chris, please, can you clarify that? And that, I think, needs to be done this way.
Well, exactly. And her comment was what I've been trying to articulate, but she did it much better. That around context of use, especially, we're all trying to advance scientific understanding. And qualification is a way to work collaboratively towards that. So your context of use may actually be what she's describing, which is proof of concept, exploratory. That is a valid context of use if a collaborative body wants to voluntarily gather science for that. So what I still challenge the group on, since that is a valid context of use, what is the level of analytical validation for that context of use? Because what's being proposed is if this is the minimum for any context of use, and that context of use is valid, then you're saying for that low level of context of use, you still have to do all 7 analytical validation steps. I just want to make sure that...
Or no, just
Go ahead.
add to that, not even just all 7, right? Actually, some of these concepts may have to be thought about very differently as well, right? Where you don't have a reference interval, right? So some of these may, we'll get into this in the next session perhaps, but I want to make sure that we are clear about these categories.
No, I think you're absolutely right. I mean, but I think there's some basic things we do for any assay, even if we didn't know, what the subject variability was, what the cut points were, right? We want to generate data so we can get there. So you need some type of validity around that assay in order to be able to answer these questions. Because again, it's just like what's in the Lee paper. It's cyclical, right? We learn and confirm, learn and confirm. I mean, it's a beautiful model.
Yep, thank you.
Yes, so I'm getting concerned over where we're going, right? Because we've been here before. We've been here we keep rehashing the same thing about being prescriptive and what we're going to specify and how a lot of it can be freewheeling as we move on. I don't know if you guys have been following, and I made this point at APS, have you been following the reproducibility crisis in science, right? It's being publicized across our field, right? There are people that are throwing out 10% numbers. There are people they're throwing out 40% numbers. People can't reproduce our research, okay? And I'm going to tell you that's a biomarker problem because all this data that we're trying to reproduce is biomarker data, okay? So I would say be very cautious even early on to say that we can all just freewheel and make up parameters as we go. I think that there's a strong argument here being at least slightly prescriptive even early on, okay? And so just kind of keep that in mind that I feel like we're kind of moving away again from specifying things. People are going to look to this paper as the gold standard for reproducibility in our assays. And I'm telling you, this is the reason why we're not be able to reproduce our data, because even early on, people are like, well, it's good enough, and how much do we really need at this stage? And the differences could be plus or minus 1,000%. So just be careful about this, right? People will look. This should be the gold standard in this paper. I do not want to say there should be no place in here where it's ambiguous on what assays are necessary, and maybe we could even put a little bit of kind of technology-based criteria in there, what represents good acceptance criteria. So,
I think the important part is identifying when you need that gold standard acceptance criteria. I think that's what we're trying to get at.
Yeah.
There is no doubt that when we conduct early assays, that's not 100%. It doesn't need to be, because we're asking questions.
You should err to the side of gold standard.
Well, no. Hang on. When somebody conducts the very first assay, there is no way that they can have a fully validated assay to be able to equal what we're going to say is the gold standard. You have to learn and confirm. It's just the way it is. It's science.
They should be able to look at this white paper and they should be able to see where their assay is weak. They should be able to. There should be no
So they
pause
should
here
know.
to say,
They should know where the gaps
good
are,
enough.
right? When they look at this white paper and they're conducting that first assay, they should know what more they need to do to
Yes,
get
they
into
should.
that standard, right?
And you have to take a stand on what is a good assay and what is not. Okay.
So, Afshin, are you paying attention? GP44, straight up, and I know this, and this may be a shock to you, there is a CLSI guidance document called C62, and I wrote a big chunk of it. It is a development and validation of biomarker assays using LC-MS technologies, which is one half of this, and it dives deeply into calibration stability, which are surrogates, to versus pools, specimen stability, and order sample and post-processing stability, CLSI C62, and on October 14th last year, CDRH recognized that as an appropriate analytic standard for endogenous biomarkers with LCM-SMS, and there is a ton of stability, and I think you owe me a beer.
Okay. This is Washington. I'm going to have my lawyer call your lawyer, and then we'll settle, and I'll leave it as that. That's all I'm going to say about CLSI for the rest of the day. Go read it.
C62 is one half of what we're describing, and I would reiterate.
It's being recorded.
I got it. Secretly. I got it. Recalcitrance isn't appropriate. I got it. But it does deconvolve a lot of what we're beaten to death here. These things have been done. I actually had a different question for you, GP44C62. I'm kind of interested on, even though it was a manufactured assay and as a system, you didn't really point out which one of the columns that you followed through on validation. You pulled data out of the table at the end of 20 days, 2 replicas. Could you just maybe deconvolve whether that chart that you showed was actually mapped on top of your timeline for me? You understand what I'm asking?
Yeah, I don't have it here. This would be actually a good way, because that would show what was done when, right? So what you hear, what I showed, the table with, let's go back to this one. This table, it's basically the status now, okay? And that status now, it's kind of a collection of what we have done over time. Right, and so I don't have that, but Shelley can comment on it. I mean, we had a quite comprehensive validation on from the get-go, right? You know more about it.
So we started way back, like I said, with the preclinical validation. The assay's the same, and we had pretty set criteria, kind of the standard precision, 3 or 4 days. Kind of most of those criteria, probably not interference, and a couple of those other ones. And then as we moved on, we went from preclinical to clinical. We added more precision data, and then we went to the LDT standard. We added 20 days and method comparison and all interference. And again, interference depends on what population you're looking at, but we added the typical ones that you do for serum assays.
I'm going to push one step further. Did you try and force failure by force lot change, force calibration change, and induce, in a short-term time frame, a lifetime concept of complete reagent switching in your validation studies?
So no, not in the validation, but we've done that within the lab because it's been so long now. We've been running this on clinical trials, but no, not within the validation.
Congratulations, because again, one of the things that I think Afsha made a point, we are drawing to a phase 2, phase 3 axis of evidentiary data. That's where we're sort of dialoguing, but we're trying to look backwards. But let's remember that we're doing this pinch point to phase 2, phase 3, because after that, these assays and these assay systems can live in clinical medicine for 25 to 35 years. So it's the time concept, the control over time that the clinical laboratories and the CLIA labs have really got nailed. And I apologize, Afshin. I'd thrown out these numbers because these problems have been solved, yes, in a subtly different way, but it is still control of precision and control of drift over time in the morass of humanity coming in and coming out of a laboratory as an example of where the clinical laboratory has really derived this framework. And I want to acknowledge the fact that you thought of that.
Yeah.
And the thinking exercise that you talked to, Jiri, I want to compel other people to sort of have whatever glass of whatever it is you drink and have those thought exercises. What if? What if? What if? Because it actually becomes a fascinating journey of discovery.
You need appropriate QC across the whole journey.
Yeah, and then also if you look at the data over time, right? So we'll look at the historical performance of the assay over the years. So it's evaluated the way for trends and if there's a drift and something like that, we would hope to see it.
Aware of the mega pools? So the industry worked a few years ago with a company called Golden West Biologicals. Me and Andy Hufnagel wrote a paper. They make 400 people pools every 3 months and have been doing for the last 4 years. So the size of the pool normalizes to the marker concentration because it's 400 people. They make them every 3 months, so you've got 4 time studies or stability studies banked per year, and they're about 4 years old now. So that is another way. The size of a pool controls the average marker concentration as a longitudinal control for drift. I'm going to drop.
No, it all makes sense, but again, right? So we're talking about qualification, and then we're talking about the clinical application, which is a little bit different. And I think we need to kind of keep that in mind, because otherwise we just kill ourselves if we're going to do everything like GLDH at this stage, right? So we need to kind of But I think what GLDH is good for, it's the example of it for qualification and for application in clinical trial, that we can see on that one example the difference. And being able to consciously say, so, what I need here and what I need there, because in end effect, I would like to put it in a clinical trial. I would like to make a decision. I think that's the holy grail, what we are going after. So, Steve, please.
Been here so long, I think I forgot my question. You actually brought forth a can of worms in your talk, and John literally took the bait by going to zero-order kinetics. So looking at this as an enzyme activity assay, I'm going to open the can of worms and let them out now. The difference between a mass assay and an enzyme assay is a very serious one in terms of how this is performed. But you mentioned that there are endogenous potential inhibitors to GLDH, as there are for our liver transaminases as well. We measure those by activity rather than mass because we know we can have denatured material. But if we measure activity in the presence of an inhibitor, we're going to have a false positive result with potentially serious consequences to the patient, right? So, was anything done? What are your ideas on doing things in a, say, spike recovery fashion where you would not actually spike with the enzyme material itself, but with, in zero-order, first-order kinetics, spike with the substrate in a substrate-deficient matrix and examine that the activity is proportionally increased to the amount of substrate that you add. So this would control for the false positives. And I say this full well realizing that in our kidney safety qualification, NAG has exactly the same issues. So how can we look at that combination to reduce the level of potential false negative results by inhibitors in enzyme assays?
That's a fantastic bioanalytical question. We haven't done it, and really not. And the question is, when one would do it, it's important to characterize the assay. We haven't done it. I'm not aware of endogenous inhibitor of GLDH. I think there must be one because there are some for everything, but I'm not aware of those. We'll.
I mean, we did do a literature search, right? Right, and we looked across it, and we came up with very little, nothing.
Nothing.
Right, but true confessions, we did nothing of a similar nature for NAG in the Nephrotox panel either, and these all apply to that as well. So it's a different way of looking at how we have to do the validation for our individual situation, which plays into a lot of the questions that have been here, even though you start with a manufacturer's platform and an appropriate IVD to do this, you still have to validate that that is working in the situation at hand in which you're working. So whether or not you have a kit to start with doesn't matter. I'm not so sure there's a 95% difference here in how we do these things. We still have to take whatever it is that we're working with and show that that meets our needs for the context of use. And that might be a lot easier with an IVD than anything else, but it still has to be done. And so I think we're all in the same boat at that point.
Yeah, I mean, how did we figure out the ALT inhibitors? I think what happened was after those compounds were dosed, we saw all zeros across ALT.
That's right, and you had
and then we
denatured
did an
ALT,
investigation,
which
right.
was inactive, and you had to go from mass assays to activity assays to be able to show that you were actually releasing ALT, right?
And the vitamin B6 was that the people they eat the processed rice, they have a low level of ALT, right?
Mm-hmm. Mm-hmm.
So it's all post-marketing. I don't think anybody.
Yeah, and that's the question: is how do we? I'm sorry. That's how do we handle that in a qualification? I mean, we're not going to have all the answers, and so I think we need to do the best we can. And
we need to think about the risk, right? So we're adding this on addition to everything else that we're running. So we don't have GLDH. These kids don't have a way to monitor liver injury. So we're not really risking these kids. And we're probably, if we get a high or a low, we're going to look at that value. We're not just going to stop dosing immediately or.
And I think another solution to it is really, monitoring your data, right? You got your limb systems, and you look at your performance of that, while you are in production over time, and
Retest.
and that way you start seeing that, like. You start getting people from like the ALT that they just were on that rice which was processed, and you start seeing trends in those, right? And then how you get to it. So I think it's important to really kind of the whole laboratory, how it works, how you apply all process is important for that.
I think that's an incredibly important point, and we touched on that yesterday, and I'd like to recapitulate that just because it comes off a CLIA platform and it works really well doesn't mean that you can't not look at the data. You still have to examine the results as they come out and make sure that they're trending appropriately, everything else is going on. And that's the part of this that we really need to put in place for looking at these things longitudinally, right? So you said January, June, July, or whatever months you pick. Are they working the same? Are we doing the same amount of good for the patients in the testing at both times? So we can't afford not to examine each one of those when they come out. If you have other reference data to base a judgment on, you have to use it and look at each and every individual point.
But again, we need to, I totally agree with this. This is really important for the application. But now, if we talk about a qualification by biomarker or validation of an assay for a qualification, you may not have this whole wealth of data and everything. So I think
know that. I know that.
So I think it's important to say what really, because the qualification is one endpoint or outcome, right?
Mm-hmm.
So which gives you a confidence into applying a biomarker in certain contexts of use. Right, but then I think that's you need to have an assay validated for that. But then you take it and start applying in a clinical trial for decision making. Then you need to have all these things in a control in order to be able to rightfully apply it. But I don't think you need to have it a priori for this kind of a comparison over time and everything for that qualification time.
But the more,
Julian, what do you think?
Yeah, but the more robust your analytical validation is ahead of time, the more confidence you can have that you're not going to run into problems later on.
Absolutely.
Right?
Absolutely.
Absolutely.
Makes
That's
sense.
it.
All right. Who's next?
Well, I'll be honest, I'm pretty woozy. I'm going to ask the organizers if they can maybe get those Friday buzzers so I can sit in my chair for 20 minutes, and when the buzz is red, I'll come to the mic. Okay. Takes a little while to sink in. I know everyone's not a gem. I'm your worst nightmare. I'm another Russ, sorry. I want you all to relax. I have nothing to ask. I just want to make a couple statements, and people were telling me yesterday I was too quiet, and I was quiet because you guys were doing a great job at arguing. But that side of the room is just terrible. I'm sorry. Okay? You guys, a pleasure to sit with. I want to make sure that, again, it's going to be based on the same statement I made yesterday. And I did forget everything, and Lauren helped me remember, and I wrote them down. People will use this paper to see where their assay is weak. Use this to drive what they do. Use this to understand what more they need to do. True for a qualified assay. I want to make sure that, again, we didn't sneak back to the exploratory, hypothesis-driven, because this is not the paper I would give those people. I would give them Gene's paper. I would give them Crystal City. Pick the number. I can't remember which one. One of six, probably six, and probably a paragraph or two out of the next one. But again, you got to separate those areas. And 44C62, the guidance that Russ didn't fully read, I think that's a great one. I didn't read it, but man, if you read most of it, that's fine with me.
I wrote most of it, mate.
So again, really, whatever you're doing, and when I'm watching this and I'm watching the other kidney ones, it all makes sense to me. And we need to do it. Patients are involved, but it really doesn't apply to when I need to put 12 or more biomarkers into an oncology study, and I'm going to use them once. Maybe I'll take a couple to the next stage. I don't even need a plate reader sometimes. I could just hold that. Yep, there's yellow there. And that is a hint, okay? So again, just really focus that down. I'm concerned that this paper has a sneaky way of showing up in the guidance document for bioanalytical method validation for biomarkers, and that scares me a lot. And now I'll sit down and your Rust nightmare is over.
Not quite. So I would ask, though, and Russ, points well taken, but I would ask to consider that there's an I in community, there's no me in community, but there's unity in community, and anything, I know, pretty good, huh? And anything that even in early discovery, if we can get away from 4615 or 4620X mindset and just generate a little bit of this intra-individual, this biological context or biological result data to just support, to just guide, as just quality information. If it's technologies like mass spectrometry or well-qualified standard, or even a standard that somebody else can buy to rebuild an immunoassay, we can provide some continuity for the growth and translation of biomarkers. So I would just ask, and I agree, it is out of scope. The exploratory biomarkers is not within the scope of this document. This is a pinch point to phase 2, phase 3, kind of. But anything that we can glean as a community to help in that translation is truly value added. And I really, really would like to see CVI, CVG, and that concept a little bit baked in early, so we can work off each other's strengths.
What we've learned is that he didn't get my joke. So I really don't hold an ELISA plate up and look at it. 4620 doesn't even enter my thought pattern, okay? The assays are developed fit for purpose, those that need it, 4620, those that need biologic variability, which is pretty much every single one we do. We always look at biologic variability. We look at biology and stability, variability, analytic stability. We do it all. My concern is it goes to the nth degree here. And if I have a biomarker in an early oncology study, and even though it's an early biomarker and in an early study, if it's going to be a go, no-go decision, that assay is more looking like this, because I know I'm going to have to kill a project.
Well, so that's a very, very clever point, and that is risk return. I've had the fortune to work at a couple of great pharma companies, Lilly in my career particularly. More work, make a kill decision, but make it smart, which is, I think, what you just
Exactly what I
alluded
said.
to, Russ.
Yep. Okay, thanks.
Like Steve, I kind of forget what I was going to say. But two comments. First is a continuum of what the Russes have been talking about. I think everybody really was responding well to the idea of the continuum of the context of use and the assay validation, but I did want to echo what Russ Weiner said in that very clearly articulating that there are assays that will never enter this continuum. And I think where I'm seeing confusion in the room is at the exploratory stage, you often have biomarkers that you can see if it starts to hit it out of the park, absolutely, that's where you're going to want to go. And so that's an exploratory marker you'd put on this continuum in the exploratory space. But then there are a whole bunch of other exploratory markers that have no interest in ever going there. They're for internal decision driving. And I think if there's a way that you can make that really concrete in the paper so people are really clear, and I can give sort of a related example. So we do a lot of target engagement assays early in development. We want to convince ourselves to make more spend on our drug. Yes, our drug got where we wanted it to be. That's a go, no go for our program internally, we're done, right? So that never needs to go anywhere near this continuum. But you can imagine a scenario where we might say, not only do we want to de-risk that decision, but we actually see there's potential that the extent of target engagement is maybe going to inform what our dosing paradigm is going to be and could ultimately be a tool for physicians to use to determine when to redose. Well, that's the same assay, but that's an assay that you're thinking if it's going to go that way, it's going to get on this continuum. So I think if we can be really clear about that, because those of us who are worried about creep are worried about those assays that we'll really only ever use for ourselves, that we're never going to ask anybody else to ever reproduce because we're making our internal, our risk, our spend decision, should be clearly excluded from this, while also emphasizing the great value of if you see that you have a biomarker you may want to qualify in the future, the earlier you kind of get on the train, the more you save yourself time. So first comment, and then second comment quickly, which will come up
quickly respond
later.
to that one, Lauren?
No, you can't, because that was absolutely logical. So no, you can't rebut. I'm teasing Russ. Of course you can.
I'm feeling the love. Thank you, Lauren. I want page 5, the first introduction, 4th paragraph. It says, clear one end of the clinical continuum, and I'm going to quote, as well as assays used for measuring exploratory biomarkers in clinical drug development are outside the scope of this document.
So, yeah, Russ, I read it. I completely agree. I think the point that I'm making is you really can't overemphasize that enough. And so if you have a figure that has this continuum, another statement that says this is a continuum for biomarkers you envision going down the qualification route, reminder, this isn't for, come on. So we all know how to present. You tell them what you're going to tell them, you tell them, and then you tell them what you told them, right? In this instance, I think we can't be too careful about that. I think anywhere you can embed in a figure, anything for your own purposes would never have to go past point X. I think it can't be overstated. Yeah.
So I just want to emphasize, I wasn't arguing against this continuum. I'm not saying that we need all validation all the time. I liked how the white paper handled this because for me, it was a progression of quality. The only thing that still worries me, though, is are you willing to publish this early data that you generate? That still bothers me a bit. And so I was kind of hoping that we would at least leave some cautions for people because I know that data is all being published, right? Oh, this early biomarker data, people are dying to put it out there. And just again, I am not arguing against this progression. Okay, I believe in it. I liked how you guys handled it in the white paper, and I agree with you.
So I hear you on the publishing. I mean, in my personal experience, it's not the people in this room that I have difficulty reproducing data. It's all the academic labs out there, right? I mean, that's the truth. So
may I just clarify what I heard from you, Lauren? You are not necessarily saying when we take internal risks and when we make decisions on target engagement, your assay does not necessarily fit that purpose, right? What you're saying is it is a quality assay. You can stand behind a scientific publication. It may not address all of these criteria to the T, right?
Exactly.
So I think we need to be very careful because I think the quality of the assay is not what we are questioning. It's the extent of validation. It's very different.
Well, and whether we want to get on a prescriptive continuum with it. That's all. The second point, really quick, because I'm sensitive to the fact I've been standing here a bit, is around the examples that go in the white paper. I think are really helpful, but also given the historic context and how long it took to generate those data to get those examples in the white paper, some of the practices are going to be inconsistent with what our recommendations are for approaches. Like, for example, when it comes to all the analyses that should be done with endogenous analyte up front versus a lot of spike recovery experiments that were done. And I don't know if you could put any sort of additional text about if it were 2017 and we were starting this same exercise of this qualification of this biomarker, it might look a little different in these particular ways.
I think that's a really good point. I think the other thing that we need to talk about also is the fact that the expectations of the FDA have evolved, right? And so, I mean, you can see what a large validation that kidney safety biomarker validation was. I mean, soup to nuts, basically, right? And so the question is, if we'd approach the FDA today, where would that conversation go? It might be very, very different. And so we need to capture that. Thank you.
So just one follow-up, one request. So to page 5, just because I like playing with Russ over there. So I was reading this introduction, and I'm like, yes, it doesn't apply to the vast majority of the work I do. And it actually says that assays and measuring exploratory biomarkers in clinical development are outside the scope of the document. And then fear went into my blood. However, the general analytical principles outlined in this document for biomarker assays may also be applicable to biomarker methods in clinical development of pharmaceutics. And that's the key, the line that gets people to go, So I would consider removing the however and moving the paragraph that's pretty much towards the end of the introduction up front. So when people are reading it, before they start reading all this, they know what the purpose of the document is. It's for these qualified biomarkers and not exploratory. And then the rest of the document is beautiful. It was a good read. I enjoyed it. And being in the companion diagnostic space as well, lots of it applies. So thank you.
Send us your red line.
You got
And just to clarify
Sorry.
my comment, much like you had done, I'm all for trying to have reproducibility of the data. We have to have that. But in the biomarker qualification space, we also have a lot of academics coming to us. And for areas where we have drug development need, for which we have no standards at present, vascular injury as a safety marker, we have to somehow get ideas out into the space to have them be used. And then from there, we can decide, do they have value, and take it to the next step. Those academics, in this case it's not academics, but we do have others, they have no intent to market, develop an assay, but we have to give at least the drug developers that want to use those markers enough details about the assay that they can at least voluntarily incorporate in their programs if they see that there's value for it. So all I'm questioning is, this is not my area of expertise by any stretch. All I'm saying is when you put forward these principles, just try to put caveats around them so that for the pharma device person, an academic person could still do this, maybe not with the same caliber or rigor, but at least make it an attempt so that we can get more information.
Again, Chris, as you know, I mean, we have the letter of support process, and with that letter of support process, we also put a data package together around that, right, which includes how the analytical was done. So that's what we're trying to do, is how do we push the community forward?
I think there's a question.
Can I make a comment to this? Actually, I disagree a little bit with what was said before, because I actually especially like the introduction as it is written, because I mean, if I start with an exploratory assay and I have to think about how I'm going to do precision whatever, and then, you know, if I have a guidance for not this assay but for a different one, the same principles can be applied. So general rules like no spiking, la la la. So this is so much easier then. I don't have to reinvent the wheel, but I just can take this out, what would apply then to the assay, and can just leave out what I cannot do because samples are not there, blah, blah, blah. But I can just take out whatever, you know, is suitable for me. And the other point is, if the selection of an exploratory biomarker is made because we think something is there. So you would never, ever select a biomarker to test, even though it's exploratory, if you don't think it could show you an effect. And what is so different with the so-called valid biomarker at the end? You want to show that this biomarker makes a difference, and this is what you use it for. So why not applying things as they are? And I think, to be honest, I like the introduction as it is written.
I want to comment. I think previously some comments have already been made that the way that we start the discussion of biomarker in more than 10 years ago, and so we actually have a group met over 18 months of time and craft out this white paper that was published. And it has all the details of how to do it, start from the very beginning. So I really encourage people who have not read the white paper, please apply it, because I also have met other people who came up to me and say, I used that white paper to follow through and it works for my exploratory phase. And also they understand it's a continuum, and then they can go to like pilot study and gather some more data and information, and also endogenous sample. So that sets the stage for now, for the qualification phase. So I hope that we don't get mixed up what is a proper use of already a white paper. And also not only that, there are many, many publications that cited in this white paper that you can use and learn from that.
This was perfect. I was actually about to ask Dr. Lee, don't leave. We have it. So the question I was going to bring up, actually a comment, was that I wanted actually Dr. Lee, that's great, you just came. Dr. Lee and John Allinson, some of the people that were in the original white paper. I'd like to hear from you guys that with these documents, other documents which I'm not gonna name, were out there, right? Why did you guys actually put the original white paper together? What was it missing for the drug development world that you guys decided to come up with this white paper, right?
Well, actually, to tell you the truth, I was the president of Clinical Ligand Acid Society. I really try to encourage the collaboration of the clinical system. And I approached them and I say, your machine is wonderful. Can we use your machine? And they say, it's a closed system. You cannot. And that's really how it started. Ron Bowser and I then started and say, we get no help because they did not realize there's a good market in the pharmaceutical world. And so that's how we started and have the discussion. And so we got a group and we have our AAPS, ligand binding focus group. That's how we produced this white paper 18 months later.
Yeah, I agree, Gene. I think my participation in Large was coming at it from a clinical scientist perspective. And I'd been publicly speaking and giving case studies of where laboratories, bioanalytical laboratories, following the PK guidance, were actually generating nonsensical clinical data. And they didn't know it. So part of it was actually recognizing which laboratories were doing biomarkers that didn't have sufficient knowledge, in my opinion, to be doing it, and also from a clinical perspective, where that may impact upon subject welfare and safety, which doesn't happen only with safety biomarkers. So I think part of it was to try and start that move away from the bioanalytical guidance and document what was critical and how it could be done differently. And I think we did a good job there. I think that was okay. To Gene's point, I think there wasn't and still isn't sufficient collaboration between the clinical diagnostic and research arenas. And back then, the top five of the top pharmaceutical companies in the world also had global diagnostic arms. And they didn't talk to each other. So whilst some of these systems are closed systems, we can actually still use them in drug development by opening up how we generate the data. It just means that you need to understand the systems and how you can do that. But some of those systems can generate very good PK and PD data and fit the actual document and the data to what's expected in the bioanalytical world. So we can get the value out of them. So I think this particular meeting encouraged me, and I think I agree with Roswina that we do need to be careful, and that particular paper is very much applicable to the exploratory phase. For me, it's just great that I think we've now got a consensus that we're not doing PK assays.
I was just going to, excuse me, add. I mean, at the time that we wrote the paper, the Gene Lee paper, where the DeSelva paper was being generated too, which we used for most of our PK work. And the Meyer-Sluice paper and this Shankard paper came out of another work stream that did the immunogenicity. We recognize that these are very diverse questions that we need to answer. So from the beginning, this was a biomarker paper. We weren't trying to retrofit PK into biomarker. So that we acknowledge that there's endogenous, there's a continuum. What your case study is a perfect example of the gold standard going all the way to making clinical decisions. We recognize that in paper, but we recognize that most of the work we do is with exploratory biomarkers that have an impact on the clinical study. And hopefully we say, hey, they correlate with the outcome of the patient for efficacy. And we get an earlier read that this drug is working. And if we have that good correlation, we want to present it to the FDA so that they say, hey, this is a great drug. We want you out there with it. I mean, there's motive behind this. Let's face it. And I think one thing I would like in the paper is some examples of efficacy markers rather than just toxicokinetic markers, because I think there's a overall need for a instrument-based platform for something that's widely distributed for all sorts of indications. but I think there's, when you're talking about efficacy markers, it's usually pretty targeted to a patient population, certain disease, and those are not going to be put on a platform. It's just economically, it doesn't make sense. There's not the market there.
Well, there are markers, right? They used for efficacy. I mean, you can do lipids.
Oh yeah, oh, yeah, okay. But I'm just saying that if you use some of those straight off a clinical assay, you're going to get questions back from the FDA. How does the impact of your drug on some other things? And there's a lot more. I always wonder sometimes if you have a biomarker that is highly upregulated, why a CLIA lab would be a little bit concerned that if it would potentially, there could be some carryover, and when you may diagnose a patient, the next sample coming into that analyzer, and because it's never seen this before. Just a little few caveats for even CLIA labs need to be a little careful about. I've had CLIA labs say, no, we don't want to run your experimental samples on our analyzers because we don't know what it will do to our what patients we might put in risk. So,
well, that's a good point.
so
Just one comment to add to my other co-authors on that paper. My goal when I went there, and that was 10 years ago, and we still have that picture, and it's a great picture of the group. And I think I was taller, less gray, and John was amazing. But I came in there with one goal, and that is to get the word fit for purpose to not be a dirty word. Fit for purpose doesn't mean a crappy assay. It means the assay is made for the purpose. And we go from something that is a yes-no up to something that's highly quantitative. And that paper is written exactly like that. And I was able to come back to management who before would say, no, I want a PK type of an assay, I want all these criteria. And I said, no, we're going to do it fit for purpose. And that was not a comfortable discussion. After the paper was published and they read it, they understood it. So that's kind of one of the goals that we had as well.
I guess as another author, I think what I was, I was in an early stage in my career, and we were bringing biomarkers along. There really were no, standards or ways of doing it. So I think it really was a starting point. And I think what we've seen as we've talked about this continuum now is it really does serve as a place to our benchmark for how to do exploratory work. So I think we can really focus this paper, as they've said, on the qualification aspect. My question, though, Yuri, you had, I think, what looks like a really good continuum, figure, if you will. But if you could go to that slide, back to some of the questions people have asked, at what point would you, say, address the statistician to find out, what your cut points are, and therefore that would have guided you as to what your control setup would have been, and how could we overlay, some of these key timings?
So I tell you that, and Shelley can comment on it. I mean, we did the cut points after the exploratory studies, and then we confirmed the cut points with the exploratory context of use studies. And now we are doing actually confirmatory studies of everything. We're going to confirm it. But the first were with statistician were basically done, right, Shelley?
Yeah, so for the publication for the early exploratory work, we were just looking at GLDH as a liver biomarker. We probably should have had David on board earlier than we did. But as it turns out, it's such a good marker, it didn't really change anything. But we during the IND submission, that's when we kind of got the statistician on board and kind of looked at the cut points. And now, like I said, we're confirming them.
So this was a biomarker that you identified preclinically, and then you implemented it into the phase 1 setting?
It's been around for a very long time. So like I said, the PSTC looked at it, maybe starting 10 years ago, preclinically, when we were the PST was more of a preclinical focus. And we could show that GLDH is better than ALT at predicting liver injury based on histopath. So we had a really good foundation. So yes, I guess you can say we did, but then we knew it was a good marker, and so then we started looking at the clinical translation piece.
So I'll just comment on this. People saying like, GLDH, how did you guys come up with that? It was forever here, right? So I say, yeah, that's been forever here, but nobody actually had clear context of use. When people were looking at it, they were thinking it's better than ALT. It's really not. It's about the same. It might be better in case of metabolic diseases because we do now population studies. It's really cool what you can do with this. But as a kind of a marker, it's like ALT, right? And when it comes really cool and have an impact is if you say specificity, the liver specificity. It's basically liver-specific ALT. And that solves lots of problems, even in drug development where you have these increases of ALT, which could be metabolic reasons, right? It could be a it could be a mechanism of action, could be anything, and maybe and those are all extrahepatic, right? So with GLDH you can address it. Fairly simple. So the specificity, when we got into it because of addressing the specificity, and then we kind of realized, wow, this is really interesting, right? And then you start talking to the patient advocates and saying like, okay, what's actually really good for the general practice, right? So, and then everything kind of snowballs, and now we are talking about, looking at NASH and all these things in large population study with thousands of subjects, and looks really cool. So that's how kind of the history for that marker is. So it's been around, overlooked.
So I guess the other question is, did you, using the data that you have, did you go back and compute, say, the performance standard using the cut point and the total allowable error as we've talked about? And if so, if you didn't, I guess then it's...
I don't believe we did.
No, we didn't, because the way we came up with the cut points was actually a really interesting approach. It's basically because clinicians know how to use ALT, we hung GLDH to ALT. There's an unbelievable correlation between the two, as you'd expect, right? If you're just looking at liver injury. If you throw muscle injury in, that's when you get the discordance. So that's the way we came up with the cut points, and it's a really tight assay. We looked at the difference between the intersubject variability, or the variability in general, between ALT and GLDH is tighter. So we need to go back and do that, just like we need to go a little bit further with the kidney safety biomarkers to do the same exact approach.
That's why this framework, right, which we are discussing, is actually important. Because, and I agree, I don't know who said it, it's just kind of should be written the way that a scientist, biomarker scientist, not necessarily an expert in validation, can read it, enjoy it, and know where to go, whom to talk to, and how to do this. So this is extremely difficult to write it the way that it's readable for general audience, scientific audience, provide enough information, not too dummy down, but it just kind of make people think that framework, what do I need to do in light of all the things with context of use, well, I have all these things what we are talking about, and validate, characterize the assay. It's characterization of an assay.
All right. Sorry to hog
Makes
the
sense.
mic. Last question.
No, sorry.
So now if I want to bring forward a GLDH, as it's already, I'm assuming it'll have a context of use application, but I don't use it on the Siemens platform. I just chose not to do that for whatever reason, hypothetically. Now, do I have to, again, is there a comparability approach that I can take, or do I have to go through and do, in essence, demonstrate context of use all over again with now the new test?
Perfect. Thanks, Shashi.
Yeah, in our qualification determination letter, we do say, or the guidance, we say that alternate assays can be used. We are qualifying the biomarker, not the assay. That said, when you use a different assay, you may need a different cutoff and so on. So we realized that, and so we actually have copped out and said that talk to the review division and deal with it in that context. But generally, I think you can address this a little better. But something like a bridging study might be the ticket. But basically, that's what we say. Talk to the review division, ask them what more is needed, and they will probably consult CDRH. And so I think.
I mean, in the end, it's
explanation
a
from
question of,
CDRH.
you want to make sure that the assay you're using is giving you reliable information. So if you're changing the platform, even if, this happens to in vitro diagnostics all the time, you'd have to internally validate that assay for your context of use for your instrument to make sure that you're still getting the same information.
And even like we're going to start looking at the kidney assays that the PSTC's qualifying, and even internally you need to run some kind of validation. So maybe not 20 days, but we would do a pretty complete validation even on the same kit internally, just so we have confidence in it.
So Please
sorry.
go.
And there's certain parameters that would be affected when you move it from a platform to a platform. You know, maybe not sample stability per se, but you know, like precision might be affected or accuracy might be affected. So you know, really, like it should be like a risk-based approach. You know, like what can you envision to be affected by this change, and then you validate accordingly.
So I'd like to say, I mean, this is a bit out of scope for the white paper. We decided not to address this because we're going to have a larger conversation around that. We, as biomarker submitters, as PSTC, as CPATH, we want these biomarkers to be used. So we're trying to come up with constructs by which these biomarkers can be used. For instance, for the kidney safety biomarkers, they're being run at PBI. Right, and so anybody could work with Pacific Bioanalytical and go ahead and qualify and use these biomarkers. The issue is when we did the GLDH, it was Pfizer who actually was a single laboratory that ran all these samples. So I don't know, you guys don't do GLDH analysis for people, do you?
Well, Shelley and I are buying the instrument and running in the garage. No, we're not doing it.
So just to further John Michael's comment, that's one of the points of the white paper example, is that all of that validation work will be published and made available, and then you can reference that. So I realize that, yes, if you need to go to another platform and do this with another test, just as Shashi said, that you need to show that it is fit for what it is doing on the new analytical system. We at least have all of those published because, again, as Pfizer, you're not releasing all of those details, and most of the biomarker qualifications that have come through already are not public. There are only a limited number that actually have data available, right? The fibrinogen one comes to mind because it was also an approved test for a different purpose. So this lets us set our goals a little bit better. If you see that all of the assays that we've used in this qualification have 15% precision every way we measure it, and your assay has 25%, you ought to be able to assume that we're going to have to do a lot more work to get it to the same place.
Well, these kind of criteria depend very much on the context of use and on the assay, right? And on the analyte that it measures. So I don't know if just looking at other qualification validation activities gives you exactly an idea of what you need to do.
No, but I think it's helpful as a starting point.
Definitely.
But
It's
should
you
not
can
be
see
exact.
the type of validation
Right.
assays, but the
Right.
details are...
So
I just want to have a simple comment to follow up Chet Ray's question. I mean, can you retroactively use the data to calculate the performance standard? And then that is estimated at the cut point, concentration, and then say that this assay is good.
Well, I think we have to. I think
in
that's
the
what we need to.
in the white paper?
Yeah, I think we need to prove that to ourselves, right? We laid this out, but I don't think we've tested it, right? And so
Right.
I think that's a good thing to do.
Yeah, we can use this example and just back calculate everything.
Exactly.
We've got all the data.
I mean, retroactive calculation is okay, but I'm just really setting, some example and say, oh, it's really useful. That's my comment.
You can also do it in the confirmatory phase.
So when I'm in this, I want to just make a very short comment on these things. I don't know whether we are overthinking it, because there is in every hospital in the clinical labs, people are coming with assays. They are qualified. They are measuring them, these LDTs. They applying them, and the doctors make a decision on it. They are governed by those whole CLIA thing, right? So now, in what we're doing, I don't know whether that is as stringent as what we are talking about it here. I think it's less stringent. Do you agree with me? Oh, good. So I just kind of think that we need to have a standard for the qualification, which gives the reviewer and us and the reviewer a confidence in the biomarker. So the assay is just the tool, and I'm sorry if I'm offending anybody, okay, to these things, but that's what we need, and we need that characterization of that assay that will allow us to assess that context of use. Then comes the application. Then it's going to be done in different laboratories. Shashi said it. We're qualifying biomarkers. You're not qualifying an assay. That's different from CDRH, which you guys, if I understand it right, and correct me, Julian, is you are basically the IVD. It's an assay, right? Which needs to work a certain way, but it's an assay. It's a different way, right? So now, if as a sponsor want to use an assay, then I can use these context of use and the characteristics, which is done through the qualification. And then I go to a lab and need to validate my assay platform the way that I can do all the work. If I want to do it for decision making, I need to do the CLIA piece on it too, and boom, I can use it. I think that's as simple as that. Am I mistaken? Okay, I'll leave it up.
Go ahead.
I'm going to go back to something you said a few minutes ago around creating the paper so that the biomarker scientists can read and understand what's needed for the validation. And obviously, the core of it, though, is for the lab scientist who's going to be saying, this is how I have to specify the validation and conduct it. I want to put out there that there are, and I'm coming from having spent many years in pharma, now being on the CRO side, there are many smaller companies out there developing drugs that want biomarkers that don't have the biomarker specialists and are coming to the CRO industry and saying, we want an assay for this, but they can't give us all of the details. And so I think this paper should spend some time emphasizing either the CRO has to create that biomarker scientist role, or somehow there has to be that input into the process for the smaller companies and the CROs that are being asked to do this. Because yes, we can develop assays for a molecule, but for this, you really need to know so much more around the process going into that.
Very good point. I mean, and I think hopefully what we can do is in the white papers do both. I mean, you know, clearly we need to speak to the biomarker scientist and speak to the bioanalytical individual and the smaller companies. It's a great point.
So I'm going to try to, bring some of the concepts that we all discuss home a little bit. And I think you're going to see that I think we're all driving in the same direction. Mm-hmm. The reason I asked the 5-6 authors to come in and talk about this great paper that they put together, right? We know as Gene Lee paper, which at the time I think I was in kindergarten, but at the time, but and I learned, right? These guys are the experts, right? They put it together, and so again, I go back to the comment I made yesterday. That's an anchor that we are using for fit for purpose exploratory biomarker. That's where we set it, right? We're all using it in one form or another. Some are more rigorous, some are a little bit less, but it's there. On the other hand, we're talking about the biomarker qualification, right? And we're trying to set an anchor right there. What I'm hoping that we accomplish, okay, there was a reason, again, why we have all adopted the Gene Lee paper. So we have two anchors. How much validation are you going to go and do to move from the exploratory eventually if you decide to go into biomarker qualification? That's somewhat company-dependent, culture-dependent level of risk. We established that. So then the question is, or not a question, a comment that I'd like to make is that whatever we end up for the biomarker qualification, let's not reinvent the wheels in terms of acceptance criteria. It goes back, if you go back to the slide that you had, that it shows the number of runs you write, or you run, right? We already talked. To me, again, it got to be a continuous process. We all agreed on that. Mm-hmm. So to me, again, this precision, your relative accuracy, the parallelism, all this stuff is there, right? How early do we decide to move from, do it in exploratory? So again, I want to make sure, and whatever we learn from CLSI guidelines, to add to this biomarker qualification, great. Some of the folks may want to take some of this information, goes back to Russ's comments that you said you remove it, but maybe we need to really either be even more clear on what we're saying, is that to move it and say, here it is. You got the 2 anchor point. Now, if you want to go ahead and use some of this for your exploratory, be my guest. Maybe you want to address that earlier. So, but just let's not reinvent the wheel at the biomarker qualification, because it's going to have a disruption in this continuous process. So I hope that makes sense. But that's what I hope that we can achieve in this white paper.
And I just want to be clear about what we're calling that 1 anchor biomarker qualification. Are we talking about biomarker qualification from a CEDAR point of view? Or are we talking about biomarker qualification for a manufactured and marketable asset? Because that's not what biomarker qualification is.
You said, see, exactly.
Okay.
Right? So
See the point of view. Okay. Yeah, it's a good point.
So just a couple of quick points of clarification, I think. I think what we're talking about here in terms of validation exercises will be very similar to what the Lee paper calls an advanced validation. And to the point of analytical platforms, I think automation in clinical labs started in 1957. Now, whilst I was around when that was the time, I wasn't working with them until 1972. But since that date, I can tell you that those clinical systems, when we think about all of these issues that can happen analytically, have all been thoroughly thrashed out. And to the point of carryover, to Jeff's point, that's a critical part of all of these. And it was interesting. There was a focus group in the ligand binding group of the AAPS called the 21st Century Lab. And they put together a list of criteria, and I know Chad was very active in this role, of what their wish list was that they would like to see on automated technologies in the bioanalytical arena. And I remember going to one of those meetings, and there was a whole list of things, and I actually stood up and said, well, you do realize that virtually all the clinical systems have had all of those for 15 years now. So it's a very well advanced. Now, I do biomarker analysis on 18 different platforms at the moment, 6 of which originate from the clinical arena. And I choose the best platform for the right biomarker for every project to get the best results. And for things like carryover, it's typically very easy. We do a prozone study with a blank directly after the prozone samples. And if that will tell you exactly what sort of carryover issue that you have. So I don't want to sort of drive regulators away more than they already are from clinical systems. I think they have a great role to play, and they can give us some great results.
Thank you, makes sense. So, anybody else? Yeah, that's an interesting question, right, Shelley? Please.
So preclinically, we've looked at like 40 plus just at the PSTC, different hepatotoxins, and it definitely depends on the hepatotoxin, probably the type of injury, where it is in the organ. So it varies. You can have examples where GLDH goes up first, and you can have examples where ALT goes up first. Clinically, we've seen it go up in the small samples that we've done, pretty much the same. And I don't know if that's just, it's more drug induced, but and minor changes. But yeah, it varies.
There is just, we are on that subject. What is interesting about it is actually it's the halftime. Okay, so the halftime of ALT, it's about 48 hours, and halftime of GLDH is about 14 to 18. So what is actually interesting, if you look at subjects with recovering and don't recovering from acetaminophen poisoning, so you can see early whether the subject is recovering or not, because they start dropping the GLDH levels much faster if they go to recovery. Anyway.
Just a last comment quickly. I want to make sure maybe we nail the nail on the coffin with this reproducibility conversation. Yeah. so again, let me use the example. If you send the same reference material, okay, same reference material that we buy from the same vendor, and you send it to the 10 pharmaceutical laboratories and to 10 academic laboratory, all the 10 pharmaceutical labs going to get it the same, almost very similar. 5 out of the 10 academic lab may get it right, the same as what we've got, right? Now, you send 15 different or 20 different reference material to all of our laboratories, we're all going to get different results. So I want to make sure this idea of reproducibility that we talk about, we heard yesterday, some today, that it has to do, we need to, worry about our people, all that. I mean, part of it is that we all have system suitabilities in our companies. We hope we have, right? The people I work with, they have system suitability. So there is a training program, there is other things. So it is usually not the analyst or the scientist, most of the time, right? So the reason we come back and we say, oh my gosh, 80% plus of the biomarker data in the literature is not reproducible. It is not that because they're bad laboratories, most of them, okay, especially the pharma and biotech, when they produce those results, those biological scientists doing that work. It is truly because we do not have a gold reference standard for 90% of the biomarker. Out of the 700 biomarkers that we go after, a handful of them that have the reference standards. And that's the world we're going to continue living in. So let's deal with it and get used to it. Because if you take WHO, NIPSC, all the organizations out there that are going to go ahead and can come up with a gold standard, they put all their resources, their entire department, they can put on no more than 10 biomarker gold standard a year out. So that will take 700 divided by 10, 70 years to get it all. So again, I want to make sure that this reproducibility, it has to do truly, in my opinion, majority of it has to be the reference material that we all use.
One thing that we didn't point out overtly is across both the kidney safety biomarkers and GLDH, we only ran it at one laboratory. We did that on purpose, right? Because again, it's not about the assay and the qualifications, it's about showing that GLDH is responding. So that was a good thing we did, right? We thought about that ahead of time, and then we were worried about the reproducibility and the additional work it would take to go ahead and cross-validate assays. So that's another thing to think about when you're qualifying a biomarker. If possible, use one laboratory.
Yeah, and you got the other extreme is, and then if you look at these clinical assays on these automated analyzers, we reran a couple of hundred samples for ALT, which was measured at the University of Michigan, and we reran them in our lab, and they use the same Siemens machines, and the statistician was saying like, you guys, you got the same data, it's not possible. It's perfected to an nth of a degree, right? So then you would think that the CDRH and the FDA does something right.
Well, for an in vitro diagnostic, it's the manufacturer really that's anchoring the assay, right? Because there's a lot of in vitro diagnostic assays that do not have a reference standard. It's very common actually, and then the performance of that assay is linked to a clinical outcome. That's how it's initially, that's how it comes through FDA. And, that's really what tells you what the number that your assay gives you means in your intended use population, right? It's anchored by the clinical performance. And then the manufacturer has mechanisms in place to make sure that, years down the road and different lots and different people and different locations, they still, when you get the number 5, you know what that means based on their original clinical performance. It's a bit more difficult to do, and you don't have that manufacturer anchor.
Yeah, that's. I think it's really important this journey and understanding the journey to develop the test at the end, right? So what I'm kind of hoping that with this simple assay, somebody already makes it and so on, we can really walk it into in vitro diagnostics, the learning which we would make on the way. It's actually that's the interesting part of it, because that could be then applied. Right, so because now we can discuss here this, the CLIA and using it because we have an assay, we have an example, right? And I agree with you, Ashwin. It's just the example is too simple, maybe on one sense, but on the sense of a process, it's actually a good example because then you can think about it. Okay, when you have the proficiency testing, how do I do it? I mean, yeah, it's easy to do it with the GLDH assay. If I have then it needs to be done differently, right? But still, we have a kind of a process which at least somebody walked through and publicly, and we all see it, and then we can adjust this kind of a conversation around it and the guidance, general guidance. I'm hearing from people here talking about the document has to be readable or needs to be good for biomarker scientists and bioanalytical scientists, right? So they have to come, because we need to talk. I mean, we are all biomarker scientists, but I'm crappy bioanalytical scientist. but we have to get together, and the outcome is a biomarker or a interpretation of a parameter in light of human health. Make sense?
Yeah, absolutely. I just want to caution, and I think it was brought up. This is a beautiful, if you take this and we put it in a paper as a flow diagram, as a process, I'm okay with that. But if you're going to go in there from a biological, I'm an enzymologist by training. So I look at that and I say specificity, where's your Kcat over Kn? Where is the, I mean, there are all kinds of stuff that I can go ahead and throw at this. So what I would say is, great process, let's map it. But for example, how you set up a specificity for a binding assay versus a potency assay or an activity assay, very different. There's a reason why 90% of the clinical analyzers out there, they like to measure things based on potency and activity and not on binding. So, agreed?
Absolutely.
Let's be careful with it.
Yep. I'm seeing 5.
Well, I think we did a pretty good job on this one.
I think it was a
clearly
really good
we
discussion.
drove a lot of conversation. So maybe you guys deserve 5 extra minutes of break.
I think so. Thank you very much.
We're going to come back in about 20 minutes.
Actually, I got a statement made. However, because it's clear in my mind what we meant to have. We got to make it clear to everybody. Because it's more of a safer exploratory use. So. Yep. And internally, you make your own decisions, have your own process. And for people up in the stress say, I don't care, I don't want to see it. We're not supposed to, we don't care. That's well. So, different exploratory biomarker has no value versus exploratory biomarker that is easier to, well,
So some of the data that we're generating in the qualification similar to what you're
Yes, of course. The types of questions, the way exactly they're asking, the context of language is not just the same as a language. It may not match up with what the calculation. So some of the things
You got my email. Just send it to me. I got it right here. Somebody else has asked for it. I appreciate it. Thanks. Make sure I get it to you. Thanks. Yeah, and it deconvolves, endogenous, but it is still phrased in 4.620 space. But with the big caveat, if we recommend you go for total level error, use those as your guidance targets for all of this validation. And it is an LCMS, and it's a big document, it's like a 90-page document. I read about, I go through it quickly, but if there's a certain page I need to focus on, I go through it. I didn't write the intro stuff, I wrote all the meat stuff, the sample handling, the sample stability, the mixing studies, the spiking, the parallelisms, it's there with one half of this. We're actually working on a proteins peptides one right now for CLSI as well.
microphone? That is actually mine.
It's time to do that. All right. 11:32. So everyone, our 20-minute extended break is up and not being extended to 25. I'll do it. Okay. Thank you all for getting back and ready for another round of participation. I'd like to introduce the next session. This was put into this program at my suggestion, and I'd like to really give you a little bit of the history why. Since we've had a number of divergent white papers and consortia and groups who have tried to do fairly similar sorts of things, I felt it was very important to involve people who had been doing this in the industrial concern, generating these data, validating these tests, and have been working with the regulatory agencies over the past dozen or so years to actually have input into the white paper. None of these people were authors on that, but they are experts in the field, and their feedback on this process is actually very important to our balancing and understanding in between the industrial perspective and the regulatory perspective, and maintaining continuity with those efforts that have been done in the past. So I would like to turn this over to Mark Arnold, who has a great deal of history in doing this kind of work, and thank you very much for putting together this session, and good luck. We'd like to hear it.
I almost feel like a Gallagher audience at this point then, that, we all need coveralls for what may be coming at us. Well, with Steve saying that he was the cause for us to be here, I really also want to thank the committee because they recognized that they were working as a closed group and they needed the industry input, and as well as having this open session and really soliciting a lot of input from the community, which I think will go to make the most robust paper possible. So with that, I will allow the rest of the panel to introduce themselves.
Lakshmi Amayawadi from Sanofi Genzyme, and I've been working in the biomarker space for more than 15 years and looking forward to the discussion.
Chad Ray from Zoetis, and likewise, I've been in the field for roughly 20 years, almost the whole time in biomarkers in some sort.
Lauren Stevenson from Biogen, been in the field for 12 years and biomarkers for the last 10.
And Steve Lowes from QSWES Solutions, 25 years in regulated bioanalysis, largely coming from the LCMS angle of things. So hopefully can bring that perspective a little bit more out in this discussion here.
Okay. So moving on. This group all had the opportunity to read the paper ahead of time and had a couple meetings, and we've collected some of our thinking here about the points that have been discussed pretty thoroughly, but we want to emphasize and then kind of use this to set the stage for any other questions coming in from the audience here or the audience on the web. And we really would like to have more audience on the web participation in this portion, because this is the opportunity where industry is really getting the opportunity to give that feedback. So please, send your questions in. Let's get them and allow this panel as well as the rest of the audience to provide input. So, we all went through the paper. We thought it was a very good framework. We liked these aspects of it, that it's framing what we're looking to do. It's giving us that context, and it's telling us not only how it's going to be used, but what the expectations are for the assay. And so the critical topics are there. It gives you the analytical characterization. The point that I like the most, which is what I championed when I was in pharma with my group and now that I'm out at a CRO, is you really have to understand the biology of the biomarker. And from that, you can build the assay specification. And it is going to be an iterative process as you go through things that you learn. You take that learning about the biology, because it may be different between healthies and patients, and you go from there. So in this case, what we really want to do is have that framework and then be able to apply it to each new biomarker so that it is answering that context of use question. We were pretty much in agreement around the scope of the paper, confirming disease modifications and limiting it to the qualification of a biomarker for a disease. A lot of discussion has been here today on, and yesterday, what is that scope? And what's the creep going to be? How do we keep the exploratory biomarkers from having a rigid framework or rigid requirements? And so I think the scope setting, we're all in agreement. That's the way to set this. Later on, we can figure out how to apply it to the other space. And then keeping it to the two technology platforms, we think it was the best approach right now. Those are two of the ones that there's a lot of experience with and a lot of opportunity. The overall strategy that you're going to go through for the validation is going to be similar. We do want to point out, and it'll come out later, that there are some differences in the types of testing that you're going to do just because of the technology difference. So with that, we're going to start going into a little bit more of the particulars, and I'll turn it over to Lakshmi.
Thanks, Mark. So I think what we're going to do is to just briefly take different points in the document that we thought we should highlight where we agree and some that need clarification, and we'll go through that very quickly for a few minutes, each of us, and then we'll go to the discussion. So we appreciate the context of use concept. Some of us feel like we've been doing this already with fit for purpose. They're sort of this one and the same. However, certainly as Russ, I think, alluded to, fit for purpose was somehow seen as a dirty word by some. So we are very happy, and we want to associate a positive connotation for context of use. This means what makes sense. And it makes sense for us from a drug development perspective for other areas of biomarkers as well. So we appreciate that. And we also, maybe touches on Mark's point, it's important to understand this before launching into assay development. So I personally don't really like to think of us into buckets of bioanalytical and biomarker, or whatever we want to call our scientists. I mean, these are biomarker scientists, and if you are indeed separating the two, you want to make sure that both parties know enough about the other component so they're able to do the right thing. The iterative process of analytical versus clinical validation was well highlighted in the white paper. And it was highlighted that these were very distinct. And I want to make sure we also clarify that they're interlinked. So this is I think it's clear to us based on the discussion, but I think it's important, goes back to the impact within our companies even, right? So you don't want to silo people who do the assays versus people who think about the biology, then you don't get the right result. And we see this internally, as well as I'm sure I've heard from some of our FDA colleagues, and that's why they're emphasizing thinking about analytical validation up front before you just go all the way through. So I guess I spoke to the next point. And then coming to the specifics of the paper, full validation versus partial validation. While I understand what it means, or some of us feel that this makes sense, I also want to highlight this could be misconstrued, where when you go into the industry and organizations that are very big, there are a variety of stakeholders who get involved, and it's a cross-functional effort when it comes to biomarkers. And there's a notation, just like fit for purpose, full validation means that's the gold standard, and that's what you have to do. And if somebody else, some other department is offering to do full validation versus some other department does partial validation, that maybe isn't good enough. So I don't know if there is some other term we can use. I think there was a translational validation that was put in the white paper, something like that in the table 1, and maybe something to think about. And then table 1, this came up yesterday, and I was thinking about it last night, and I looked at it again too. I think it is causing confusion, especially in the exploratory section. While we understand in the white paper it means exploratory phase of biomarkers that you intend to qualify, there's a whole bunch of other exploratory markers, as many of us talked about. The 90% of the biomarkers that are being worked on in the industry fall in this bucket that are internal decision making. It's clear that that's not what you're referring to, but I think it's important to, we cannot underestimate the importance of highlighting that as well and make sure that that's appropriately covered or taken out altogether in the table in the exploratory aspect of it. So specificity. So I do want to mention, when I was looking at the paper, there's a section there. It talks about for large molecules or LBA, ligand binding assays, that's not possible to achieve. I think Afshin talked about this as well yesterday, that, you know, I want to make sure people don't walk away thinking large molecule assays do not have to be specific or you cannot achieve them. I think it's worth clarifying what we mean by this. I think irrespective of whether it's large or a small molecule, it's essential to know what we're measuring, right? So we have to consider the context of use again. What is the specific molecule you're trying to measure? Maybe there are multiple isoforms that your assay measures, and you don't care which isoform it is that you want to detect, then that's okay. So you're making a conscious decision that you don't need to measure it. For example, the IL-17 story, right, with IL-17A dimers, heterodimers of AF versus homodimers of FF, which ones do you want to measure? And maybe you don't care, or you do care, and in which case you want to make sure that your assay is specific. So I want to make sure people don't walk away thinking you don't need to think about it. And the way you achieve specificity with the large molecule LBA is how you generate your antibodies, and you screen for what you are looking for. So sufficed. Next slide. So PK assays are not equivalent to biomarker assays. So we're all on the same page on that. I think when you read the paper, and as you know, Russ Wiener again referred to, there are places in the paper that keeps going back to this PK assay validation. Many of us prefer that we go at it de novo, thinking, as fit for purpose, paper did apparently, I just learned today, de novo thinking about how do we qualify or validate these biomarker assays. I think that would flow much better, and we would eliminate a lot of the controversy. If some of the aspects of CLSI guidance apply, some of the aspects of PK apply, please incorporate them as fit, but don't have to keep going back to them, other than citing them in the references would be one way to deal with that. Again, going back to the risk of misinterpretation, I think someplace in the paper it talks about starting from the PK assay and then sort of working down from there. So the reference material versus calibrators, or the calibrator rather. So the paper did a good job of highlighting the limitations of protein biomarkers and how to address them, these differences between reference material and calibrator, and how you address that in your assay, the limitations of it. As we alluded to yesterday, the term and the practice of commutability is a missed opportunity that hasn't been addressed in this paper. I do want to highlight within AAPS, one of the subgroups, which is dealing with the biomarker discussion group, there is a paper that will be coming out soon. It's accepted on biomarker protein standard identification and characterization. And that really has a good deal of relevant information, including number of case studies. Again, as somebody pointed out earlier, 90% of the problems really with the biomarker comes from the fact that you may not have the appropriate standard, and how do you deal with that? There, they also address the commutability using the statistical approach of Deming residuals, and I don't want to go into that, but I want to make sure at least we highlight that and that can be referenced. So when calibrator does not equivalent to endogenous analyte, right? And that's when we want to go back to endogenous samples, because that's the biomarker you're measuring. And assess your validation parameters, analytical validation, based on your endogenous samples. So rest of my colleagues will address each of these parameters as relevant, but I want to talk about stability very briefly because that won't be covered later. So stability, I would caution that we give a hard look at using endogenous samples for stability assessment. In the table, in the paper, it does talk about reference standard or endogenous sample. So I want to make sure that people understand the liability associated with not using endogenous samples. And I want to quote Stephanie's paper where it clearly highlights two different biomarkers. One behaved where the reference standard stayed stable for a year and a half, where the endogenous samples were only stable for 4 months or something like that. May not be right on the numbers. And with the other biomarkers, the endogenous samples were actually stable for more than a year, whereas the reference standard was not. So they were able to rescue that clinical study and go back to generate a lot of valuable data, even though the reference standard did not reflect that stability. So I do want to make sure that this paper is a good opportunity to highlight that and not avoid the misleading of you could use a reference standard as well, because that could be a potential liability.
Okay, moving on to Chad.
Yeah, so I had the opportunity to talk about statistics that were discussed in the paper. And from where I stand, I thought the paper did a really nice job of incorporating the concepts of total allowable error, which I've highlighted here. And I think I brought it up through the discussion. I think what we have to do is to figure out how to now operationalize these types of measures, incorporating statistical support, and when is the right time to engage and build that in. So I guess just a couple points at the top there. I think they did a really nice job of describing an experimental design that would allow you to parse out the various variance components, both within subject, intersubject, and also the analytical, the CVA. So I think that was done very well. The concept of bias, I'm not going to cover it on this slide because I think Steve will cover it with much more detail, and I think it'll lead to a fairly robust discussion, but purposefully use the word bias as opposed to accuracy. I guess where the panel was a bit split, not everyone may agree with my perspective on the stats, but so some of the comments that came from the panel were concerned that maybe we were too prescriptive with 0.5 times 0.25. How do we really set those benchmarks? And then what was needed, we need to take into account biological analytical variability. I think all of us were in agreement on that. It just makes sense. It goes back to the learn, confirm model that we've stated from the Lee paper all the way through this. So I think we can go on.
Okay.
Yeah, so I'll take this one. So relative accuracy bias. Obviously, there's a lot of still perspectives that remain in our community about this. It's kind of key to this discussion. This fact that you don't have a true value of the endogenous level is a fundamental challenge to the bioanalytical community that have been used to working with xenobiotic drugs. So, but that said, that I think we're starting to get a healthy discussion around it. I think we're having the whole bioanalytical community recognize the problem and come to the table to discuss all of this. And I really do hope that some of the comments that are probably being thought of and discussed on the other side of that camera there get it into this discussion. Obviously, measuring accuracy of spike standards has limited utility if it's not connected to the endogenous analyte. We've had that discussion here over the last day and a half. It's going to continue. This concept of the continuum of working from an accepted assay all the way through. That's a good one. I think it's a little bit dangerous to think that we have to set this gold standard early on. That as a gold standard, it doesn't. You can't you've got to do better than all of the time. That seems to be a conflict of terms. If it's a gold standard, then it's a gold standard. But we're talking here about establishing something that we can build upon as we go through this continuum. So I hope that is preserved. Relative accuracy or bias being linked to the context of use in itself precludes prescriptive criteria. But let's also keep in mind that it's got to be relative to something. Even with an LCMS assay where we're dealing with a well-characterized reference material, it's relative to that white powder. So it's always relative. So I don't think that should be something which scares the bioanalytical community. We just have to think about it in a little bit of a broader sense. We've mentioned that commutability isn't addressed, I think, well enough in this paper at the moment. And it is in itself, I think, predicated on having a decent handle on accuracy. Without that accuracy, how can we be able to say that we have an assay that we can compare to something else? Knowing what it's relative to is the fundamental aspect of that commutability. Defining accuracy bias of a bioanalytical assay is a consensus, but it's developed a lot of confusion, and we need to get through that. We need to get past that, and it's a real fundamental of this discussion. That bottom bullet point of protein versus small molecule biomarkers, I would suggest that we need to, if we're considering LCMS, and in the white paper as it stands at the moment, there are certain sections that make reference to the fact that cases where we do have a well-characterized reference standard, then let's take advantage of that. I believe that there's a lot of the bioanalytical community are now working with biomarkers where we do have a well-characterized reference standard. And when you have that available to us, to take full advantage of that. And I think that that needs to be fleshed out a little bit more because there's too many of us working with lipid biomarkers, with peptide biomarkers, where we can do a very good, thorough job around getting closer to that absolute accuracy concept. So I'll leave it at that.
Okay, so I'll pick it up here with parallelism and dilutional linearity. We discussed a fair bit of this yesterday. We really just wanted to highlight here to make it abundantly clear in the paper that dilutional linearity is related to control samples, which are spiked with reference material, whatever reference material you have to hand. And I won't go into the details about how you assess it, but it does require that an expected concentration is known. So therefore, as Steve just referred to, if you don't have a well-characterized reference standard that you're fairly convinced is very much like, if not exactly like your endogenous, then the relevance to your biomarker assay is going to be more limited. Parallelism, as Steve clarified very nicely yesterday, requires incurred samples, meaning it can only be done with endogenous analyte. And that is where you want to demonstrate the parallelism between your standard and your sample dilution curve. Here, the relevance to biomarker assays is absolutely fundamental, right? I think we had good discussion yesterday where we all agreed, whenever possible, absolutely, endogenous analyte is the gold standard. So, of course, there is complexity in practice with that, right? Because often, as we spoke a lot about yesterday, you don't necessarily have samples with high enough concentration of endogenous analyte to perform the assessment the way you would like to. But you need to be really careful when you think about spiking on top of that endogenous analyte. And that was a little bit of what I was referring to and related to our case studies, is that this concept has really been discussed quite robustly in the last couple of years, especially since Crystal City 6. And I think there has been a change or an evolution, I guess I should say, in the scientific thinking here. And so what I heard yesterday was we all agreed that if that's all you can do at a given time, it's very well understood that's all you can do. But that there should be perhaps some clearer statement in the white paper that says when you do in fact have access to endogenous samples of appropriate concentration, that's something that you want to go and revisit. And I just want to turn it over to Steve in case he has additional comments regarding in the context of the LCMS assays where you may have better
With regard to
standards.
parallelism, yes, yeah. I mean, that is one point. It's where we do a parallelism experiment with the surrogate matrix. Of course, we're doing that with a standard addition, and Lauren will point out, well, if you're doing standard addition, what are you spiking it with? And if it's not your endogenous analyte, which it isn't, of course, then it brings into question what are you doing there. So we acknowledge that that is something that we have to address. But where we do have a good, well-characterized reference material. Certainly in the LCMS community, we still have to do a parallelism experiment of showing that the surrogate matrix that you're using, blank matrix that you're spiking to, behaves the same way as the actual sample matrix. So that is one example, and just at the break I was talking with Chris Evans that another example of an LCMS experiment that we can do, we have to do, is post-extraction sample stability, and that's not something that the ligand binding community are doing, but it is something which is a nuance and a well-established experiment that we do and we can do and we should do with LCMS, and it needs to be added to the discussion here. It needs to be added to the paper.
Okay, and then as I touched on earlier, but I wanted to highlight here, is that, again, the relationship between parallelism versus spiked recovery. And we really wanted to highlight from the industry perspective the rich data that we can mine from these parallelism experiments. It's not only the parallelism test itself, but as Afshin highlighted yesterday, use it to identify your MRD, and I know he spoke about Prozone. But what wasn't discussed was a means to also estimate your LLOQ in relation to your endogenous analyte. I think all the examples I've seen over the course of the last couple of days when they were speaking about determining LLOQ, that was still with a spiked reference material and not with the endogenous analyte. It would be nice to see if we could incorporate that into the white paper. And also that when you assess parallelism in multiple individuals, this is actually a really robust way to assess selectivity that's going to be directly related to that endogenous analyte as opposed to a spike recovery experiment. So really just a way to highlight that there is certainly value, and there's a time and a place when that's the best you can do to do a spike recovery experiment, but it doesn't replace the true parallelism experiment. And it really doesn't tell you about the selectivity in relation to your endogenous analyte. So if that's the space you're in, just sort of a caution to say, that needs to be interpreted conservatively. Accurate recovery of spiked on top of endogenous it certainly has helped me sleep better some nights in the past, to feel like, okay, certainly in a ligand binding assay, I'm reassured that my critical reagents are seeing my endogenous and my recombinant in a similar fashion. But I've also had perfectly viable biomarkers for measuring the endogenous analyte where that addition experiment, A plus B does not equal C, it equals, something like W. So it doesn't mean that the endogenous analyte can't be measured well. And when you do have a case where it adds up, it doesn't automatically mean everything's perfect. So that's it.
Okay. Steve, were you going to cover this one, or was I?
I think you were taking this one, but I'm happy to take it. So the framework is what we've started with here. That is, in my mind, the takeaway that we need. That we need something that that is a community, whether you're a bioanalyst, whether you're a biomarker specialist, whether you're a statistician, that we can pull out and we can use. I'm a little bit concerned with the references that we have in the document now to, say, the CLSI documents that many of us don't have access to. Hopefully something can be done about that, but at the moment, there's a lot of references to those. So Russell pointed out that there's a CLSI document related to commutability. Just go and look at that. Well, that's not easy for me to do at the moment, and worse so, it's not easy as a CRO to be able to go to my clients and say, hey, there's the reference document that you need to look at, and so we all get on the same page. So either we have to extract that out of the CLSI documents, or we need some mechanism of being able to get all on the same page. I like the concept that was mentioned this morning about this being a kind of a thought map that we need to go to. We don't want to be too prescriptive, but we do want to be able to still talk to a process with a kind of a common language across our community so that we can all be saying having the conversation. We're not talking different languages. And this point that the expectation that there's going to be this continuum, there's going to be this iterative process of moving through the pipeline of a drug. Obviously, that's something I think that we seem to have universal agreement on that there is the expectation there's going to be that continuum. And then the last context point of differentiating drug development from surrogate endpoints, I think we've pretty much hit on that one already, and I think we're on the same page.
Yep. So we're to the kind of end of our input, and just some of the things summarizing what we put out there. So it's really turning it over now to the audience here, and hopefully some of the folks on the webinar who may have sent in questions. If you don't have anything, we'll start talking amongst ourselves and discuss some of the issues.
Hi, Shashi Amur, FDA. About the surrogate endpoints, one can theoretically come to, the biomarker qualification program also for qualifying biomarkers as surrogate endpoints. So, but that's not the only mechanism, as you know. Just wanted to point out that that option is also there.
So, as world-class experts, you clearly demonstrate content knowledge. I really thank your thoughtful considerations, and I know we will certainly welcome prescriptive input, not just I don't like this section, but rewrite a section for us. We certainly look to you guys as leaders in the community to do so. I wanted to just dig into a few little things. So, Steve, we talk about small molecule biomarkers being exactly the same as the endogenous form. And I want to make sure at least people are aware that biology in endogenous binding space is different versus surrogate calibration matrix. And so even isotope dilution mass spectrometry, we do have to be very conscious of binding the internal standard, labeled form, with the analyte in a specimen. So we did downplay the accuracy piece, and please tear this apart and say you don't like it. We downplayed accuracy because the end result we aim for is a reference method, a higher order reference method with higher order, a value assigned reference materials in true human matrix as truth. That's our anchor point for true accuracy. And so we didn't want to get into this accuracy, I can spike and everything looks good. I'm a spiker, like Brad Ackerman. If you're listening, you're still a spiker. Sorry, Brad. We didn't want to really use the word accuracy because in true metrological space for us, it is reference method, but it's commutable, i.e. all of us can run this sample, commutable reference materials, which made us back off that because as we've described, as Steve described, there's so very few of reference material. So I just wanted to say that that's how we flavored it, but if you disagree, we would welcome the feedback to recreate that in some manner. I have a few other points, but Steve, I'll just shut up.
So just to comment on what you were saying, I think one of the points that you raised, it may be missed some in the traditional small molecule PK world, which is that binding event, and with carrier proteins and the like for these types of molecules. And so people tend to spike and to do an extraction for their recovery rather than spiking, doing some sort of equilibration, and then doing it. And I think with biomarkers, because of their complexity and interaction so many different ways, we have to pay a little bit more attention to that aspect of it. And whether you're using the endogenous molecule, if you have or the well-characterized reference standard that you're spiking in, we've got to make sure that's part of the process.
And actually what you just said, spike in recovery versus spike in equilibrium and then recovery, are two different things, and they should be noted as such. So they will give different answers. But actually,
Touch on one of the bias points. Steve, you go for it.
one comment on reference material and commutability. There was a deliberate omission of commutability from the white paper out of respect to our colleagues who are publishing that paper, which is due very, very soon. Having seen it pre-publication and had the opportunity to review it, there's a lot of good material in there that would be appropriate, but we did not want to reproduce that or attempt to reproduce that. And that will be certainly a very valuable reference to add into here. So when that is published and available, we will amend this appropriately to go along with parts of that we think are necessary. Also, having been at some low level a part of that, there are a number of very pertinent suggestions about reference material and commutability of reference materials, such as we can do in the biological field in there, that we would, of course, adapt into this.
So I was a big proponent of incorporating commutability, but after hearing the conversation for the last 2 days, I'm not so sure it's as important, because the way I had envisioned this process, again, would be a biomarker was qualified, and then all you needed to do as the next lab coming forward was show some degree of commutability. But I don't think that's what I've heard here the last 2 days. It's really there's an absolute linkage between the test and the biomarker qualification. I don't think, from what I'm hearing, we cannot differentiate those two. They're inextricably linked. Am I wrong on this?
However, maybe within this qualification, right, it took 8 years for kidney markers, and I do not know, what type of, issues you had to deal with in trying to bridge variety of lots over the years and things like that, right? Even within one assay and let's just say one lab.
So commutability would not necessarily be important in the first round of qualification, because assuming you're doing this at one facility, you have no need of a commutability concept to do that. It's when you get beyond that point and go to the next context of use or actually put this into a production environment that would become important. And that makes perfect sense here. And although there is a linkage between the test and the qualification of the biomarker, as we have heard, that is not a direct one-to-one link. And this is what happens when we get to the next step of how do we qualify that biomarker for a slightly different context of use? Or does someone want to use that biomarker in a different facility, and how can they generate data on that biomarker? So those are questions which we're not quite answering here, right?
And that last point is something we were discussing at lunch, especially where if you have different capture and detection reagents than the original assay, how similar is what you're measuring at that point? And so what hurdle do you have to go through to use it in that context of use?
A more detailed question is, if you have the same capture and detection reagents, but somewhat of a different system, and we know that things are different in different places, what do you have to do as well? I think everybody here realizes that the knee-jerk response is, yeah, you change your antibodies. Oh, we go back to square one and start over. And that's probably the appropriate answer. But it is a iterative process, a continuum, as to how you have to go back to do that. And our colleagues at the FDA have said that, there's likely to be similarity, but not concordance between the two methods or the two facilities doing it. And so I don't want to speak for Shashi, but I've heard that remark enough that we're qualifying the biomarker and not the test. And in other situations, you have to show that the test is suitable for the new context of use.
Or the same context of use.
Or the same context of use. And I would go back to the principles in the paper and say, here's what we think we need to demonstrate or re-demonstrate or change how it happens.
I can add a little to that. So we also described the performance characteristics of the assay that was used for qualification of the biomarker. So there is some point of reference, if you will, to see what what was achieved by that. And to be fair, we only have a qualification of 3 clinical biomarkers. 10 of them are non-clinical biomarkers, and for one of them is an imaging biomarker. So if you take that out, the other 2 are prognostic biomarkers, and for both. One was diagnostic, prognostic. But for both of them, we had FDA-cleared assays that were used. Even so, a different cutoff had to be, determined for the intended context of use. So that was the main challenge. One challenge we had with one of the submissions, plasma fibrinogen, was there were multiple FDA-cleared assays. And this was retrospective data, which was generated from others. So it did bring up a challenge. I'm not sure it addresses your problem, but just trying to tell you our thinking when that happens. What we thought we would do is we looked at qualitatively, is it going in the right direction? Addition of plasma fibrinogen, is it benefiting? So qualitatively, some of them were enzymologic assays and some were immunologic ones. It's not easy to think of bridging that kind of scenario. So we chose one as a confirmatory data set, and we gave performance characteristics of the assay that was used for that confirmatory data set. So that's how we have, it's complex. I mean, even in the world of FDA-cleared assays, it's complex. So when it's not, then I do understand. But we do give performance characteristics and what's acceptable.
So I wanted to touch on a couple of concerns you described, and one I think, Chad, that you commented on, and I think one you talked about, Lauren, which is the easiest one. So Lauren, you described the hard math or the locked-in math of desirable, minimal, and what is it? Desirable, minimal, and something. CVA, CVG, the bias and the imprecision. I just want to echo, those are really quite entrenched in clinical medicine in terms of the desirable imprecision and the desirable bias. And the bias question is going to come back in a sec. But did the language make sense in terms of why those are designed the way they are, the false error on the 2 ends of a reference population? They're designed around 2 ends of a reference population to determine if you call something normal or abnormal inaccurately based on your analytic measure. Did that come across at all in the documents or not?
Yeah, I understood it. I thought
you're an ex-clinical chemist, mate.
Well, I think one of the concerns that the panel brought forward was maybe there was too much detail, right? And so you could go to the reference, but then again, I think you need access to it in a distilled manner, and it was there. I didn't have a problem with it, so I think you'd have to ask other panel members.
So I got feedback around that. The clarification there is not so much what the content was. It was more about the placement and the extent of the content. So it may be a stylistic comment more than anything else, is that embedding that level of detail in the document in that fashion disrupted the flow of these are the things that you want to look at and how you might want to approach it. And so a lot of comments that I heard over dinner last night and in informal conversations was a lot of this material fits perfectly as an appendix. So those details are there. And the main body of this, you say you need to consider this. There's a really good way to do this, and it's right here for your reference. So it wasn't about, the feedback I heard was not about not understanding it, not thinking that it wasn't a good approach. It was twofold. One was being careful because this is meant to lead to the basis of guidance to make sure that it's not interpreted as this is the one and only way to do it, and then to provide the right level of detail in the main body and give people the access to all the details in the appendix.
I would add, just from personal experience, that is the easiest way to do it, because the other way is 120 reference population in every single breakdown of the phenotype of a biomarker. So that is the simplest published form we know about as it looks like an elephant, but it's only a small bite of an elephant kind of thing. Sorry, you had a comment.
I was just going to make a comment related to how people reacted to the positioning of that statistics section, right? The first question people wanted to understand is we place it before validation, and how many people really operate their methodologies by defining total allowable error, right? The first thing they do is to assess what the assay's position is and potentially bias. Even bias is looked at later on when there are adequate number of biological samples available to even understand whether you're generating a false positive or a false negative response. So I think the authors of the paper really need to think about where is the right place to put this, and do people really operate by thinking about how much precision can I allow before even understanding what the biological variability is. So there's a conflict in, it looks like we are prescribing that they should think about it and in the assay design phase, when really they don't even have the adequate number of samples to assess variability. So I think that is also sending some mixed messages.
Steve, I'll try and address one piece of that. Okay. So you may recall Crystal City 6, I said 4.6x is arbitrary and capricious, and I still believe that. I think we positioned something as this was a very hard section to write, I know, because I had to do it. Thank you, Steve. And we try to flavor it two ways. One is a priori measurement, and then one is how can I use my validation statistics and then get confidence in fold change. So it was hard to write. We tried to position it up front as an either or both construct, and then did really try and say, but go and talk to a statistician, because you're getting very, very deep into the weeds of noise in biological signal, and then signal over noise with noisy measurement superimposed upon top of that. So that is very, very hard to put down in a few paragraphs. We said it there. I mean, rip it a new one, I wrote it. Rip it apart, rewrite it, I don't care.
Yeah. I mean, I commend you for taking this approach to it. I think, and you'd probably do a poll here in this audience, and you probably get a lot of people saying, yep, makes sense, I like the way you stepped through it. You know what the dilemma is, though, don't you? It's the great unwashed out there that are going to be dealing with this, and that is something that we're really going to have to step them through it. And I think as long as the leading community groups, such as what the focus groups that we have in AAPS, can get this messaging out and help explain all of this through the bioanalytical community as a whole, I think that is going to be a requirement. Otherwise, to put this out as it is, it'll cause some to panic, and it can't just be a standalone document as it is without the follow-up that I think it deserves.
Okay, understood. But one of the other points behind this was actually in looking at bias, which tends to be a component of this. There's a distinct tendency towards looking at a false bias or looking at no bias at all, ignoring the bias. And by false bias, I would mean you are using this relative to an inappropriate, well, not the best standard that you have. So there is some bias that you are not measuring unless you're going back to in vivo incurred samples to do this. So again, all along that space, from ignoring the bias to having the correct bias in your assay for an in vivo sample, where can you set the error goal to go back to the clinical conclusion that you want? And that really was maybe an underemphasized part of this, but that's why this was there. Because until we understand, we're pretty good at getting the other half of this imprecision equation. We've been doing that well for a long time, but really determining what the bias is, and is this the correct determination for bias, and can we pop this into the calculations and do that, was a major part of this, because that's frequently ignored. And if we are doing this on an endpoint of a clinical context, then we have to understand what that difference in bias is, or it may become very important to those patients.
So I actually want to leverage that and point the question back to you guys as experts. So we tussled in the absence of a reference material or reference specimens, metrologically anchored, higher order reference method. We struggled with how to get you a bias number, or how you get a bias number. So we posited, individual QCs or calibrators at the end, which we're used to doing in BMV. We also posited spike and recovery as a surrogate for a bias number. But what other ideas would you bring forth as a way of trying to get close to truth in the absence of truth? Because we struggled with that one, honestly.
I thought you did it right. The three-tiered approach you had to assigning bias, that's really the only way to do it. But in an exploratory setting, those first two are irrelevant. We don't have that information, so you're left with the idea of spike and recovery. We all know what that.
Right, but
what's the
understand
value
that
of
then you're missing those two pieces, and that bias number is as good as you can get, but it's not hard and fast. It's not what you want to live with in the clinical arena.
So the
and
question-
that's really what we're saying here.
Right, but the question is there a continuum of how we can acquire this data, right? So you start off by defining error, the total error with this precision, and then you move on to acquiring bias as you are getting more of the relevant reference standards available, and you eventually end up with your, allowable error, the total allowable error, which takes into account both the bias component as well as the precision. So really in the evolution, where do we truly, which is the stage of development of the methodology are we able to assess bias? So we just need to be realistic about that.
And I think that goes to what was mentioned before, is to take this to a practical approach that people can pick up and follow. And so having, the context and things that in the body of, this is what you should be checking, but that appendix that gives you more, but interleaving it to things broadly.
I'd like to see it as one of the case studies, because I'm already confused about how I'm going to apply this, and I've been doing it for how many years, and I know people that don't have experience are going to look at it and go, "What the heck?" So it would be nice. We were talking about the value of case studies. I think a case study that shows exactly how to go through this step by step and has actual numbers would be a great deal of help. And I can't remember if the ones we have in there have this already. Okay. Do they? Okay.
You're
so
asking for
what
an
I hear from
example.
you two is that you want us to be more prescriptive in the process of how this is to be applied.
Right.
But the danger in the case studies is always that those numbers tend to become the prescriptive part of the process. And so we'll have to make sure not to have that happen, because nowhere in here did we suggest that this amount of error is appropriate or required at each and every step of the way. That has to be individually determined. But I can see the general application for a more well laid out process of how to apply this sequentially to get to the end when you need it.
I think what Mark is suggesting is sort of an example. It could be a mock. It could be mock numbers.
Yes, I just want
the
a concrete
example has to
example.
go-
I want actual numbers filled in. When you guys start, we start conceptualizing. Well, we're going to do
algebra,
total
X,
error.
Y.
We're going to use this program over here, and that's kind of the way I saw it the last time around. Did Procter and Gamble have a program for calculating total error, and everybody was using it, and we were using it, but then no one understood exactly how to explain what they got out of it, and kind of all of this. And it was the case, and so I would just like, again, simple numbers, make them up if you want, I don't know, but show the iterative process, here.
Okay.
I've decided at the very beginning on my assay, here's what my bias needs to be. And here's what I did next, and here's what I did next, and show actual numbers so people can follow through. And if they want to, they're just going to put their numbers in, and hopefully they'll get to a place where you want them to be.
Yeah, I mean, I think I said it earlier. Yuri's example should be a perfect one to be able to do that, and at those different points along the way, compute the performance standard when you knew what the cut point was, and I think it could be a really good example.
So I just have one little thing, and then I'm gonna sit down. Promise. So the table that computed CVI and CVG, we generate that as an Excel document. You can plug your numbers in and follow that experiment. Would that be good to share? It's locked into unless where you need to put in your numbers and your alpha factor, just to share that, and it just does all that
Any
calculation.
tools, I think, that can be shared with the community around any of the things that have been discussed would be valuable.
Okay.
I got a 5-minute warning. We have some people who would like to.
Just a quick aside, were there any questions from online? No. Okay.
Okay.
So did you guys see he asked all these questions and then he goes, "Do you have any question online?" Just kidding. Just picking on you. Just 2 quick comments, okay, guys. With regard to specificity, I want to make sure again clarify this, and if it's not clear in the paper, we will do it. We have obviously put specificity as an important factor, one of the 7 core factors. That's one. Number 2 is that I want to make it clear, we will never ever be able to be 100% sure about assay specificity for large molecule. And a quick example, the antibodies we generate, usually they're using recombinant protein. The second here is that even if it's against the endogenous protein, those post-translational modification that may happen, you have a different diet, you have an argument with your wife, and then you take the blood and you're going to use that too. So there are changes, right? So I want to make sure, again, we have a stress in there about, yes, go try to find, make sure the assay is specific, but the reality is we're never, ever 100% sure until 20 years from now, somebody else finds out that there were 4 isoforms and not 3. So that's one. The second thing is with the parallelism, points all well taken, Lauren. And if you're not clear on the paper, we got to be more clear. But we have mentioned tiered approach. We have put parallelism, not dilutional linearity. Well, we're going to remove it then. It says parallelism, dilutional linearity, spike. Okay, I don't think so. What we need to do, parallelism is one of the 7 key factors. Probably for my lab, the second thing I look at, even if it's at the early stages, right, to establish that. So we have basically, with this sample, what kind of sample we use. We have taken in the paper, we said tiered approach. What do we mean by that is that go use endogenous sample exactly as you said it. But again, when the endogenous sample is not there, you got to go spike on top of it. We have also included in the paper that, remember, that's the best you can do. You can sleep better at night, but that's not a true representative. So when the sample is available, go in there and as an amendment, add it to your report. So I want to make sure it's there, but the fact that you guys providing the feedback, we probably got to go back and provide some clarification.
Exactly. It was more about that because that spiking experiment is not actually parallelism. So I think it would help if it were, this is what parallelism is when you're in a situation when you can't actually do it.
Yeah.
You can do this other, which is really dilutional linearity on top of whatever endogenous is there experiment as a hybrid approach. But with regard to selectivity, I didn't see anything about parallelism informing selectivity of endogenous analyte. I only saw the spike recovery approach. So again, if that's all you can do, I think clearly articulating that would be helpful.
And the only question for the panel, there are instances, and I think we saw one example, there are times that you can actually stimulate a cell line, right? Get the endogenous form. At least you think that's the endogenous form. And then you spike it into a proper matrix. Do you consider in your laboratory as a true parallelism study or don't?
We consider it a close facsimile, but it's not parallelism.
So they still, under those conditions, you try to go back and find a sample.
I mean, I have to tell you, currently, I don't have any in-study samples like that, but I do have samples that we created in that way.
Okay.
And when we have samples of sufficient concentration, we would absolutely go back and do that again.
Great, thank you.
Okay. So we're down to like 1 minute.
Yeah, I'm just a comment since I don't know who the organizers are. So I did get a text that's they're having difficulty using the chat. So during the break, if the organizers can just check, that may be one of the reasons why we're not getting any questions. Okay. Over to you.
Thanks, sir. So, Steve, one of the comments you made about diluting out endogenous specimen with calibrated matrix surrogacy, that was conceptually in the 5-point admixing section in the document, but it was apparently not clear that you mix a low calibrator with a high sample as a 5-point admix, or even the reverse, depending upon what you have your hands on. That wasn't clear. That certainly was part of the flavoring of how it, again, subtlety, I guess. Wasn't clear?
No, I didn't pick that up as clear. I think I can maybe pick that up with you.
Right. I mean, I just want to make one comment to Afshin. I mean, since we've been talking about specificity, parallelism, I think many of us are on the same page. You did a great job yesterday with those figures with the parallelism, also showing how to accept your parallelism. But I do think in the paper, tweaking their wording so it doesn't lead to misinterpretation. Because as somebody said, 10 years down the road, you want to be using this paper. Yeah. And there'll be a fresh batch of scientists who are using this paper.
And guys, Mina and I, we were talking about this over the break. The challenge, what we have faced, is that you saw the parallelism section was this big. Then the stability is this big, right? And it's not that we're putting more emphasis on one versus the other. We were challenged as how can we go ahead and put this paper together to provide the gaps that we saw in certain areas, right? And we're struggling with it, to be very frank with you. How much detail do we want to put in there? I'm hoping that later on, as you guys were talking later in the afternoon, we provide guidance from everybody here as well as over the phone as what is the level of detail you guys want us to put in there, and what is adequate, or do we just refer to a bunch of white paper? Even the ISO, the document we're talking about, right? It's a valid point. For me to begin, I had to go in there, pay first of all, get a permission in order to get in. Then I had somebody after a few days tell me, yes, we check your credential and we think you're going to add value to this society. So then I get paid a fee, and then I had to go in and pay for it. So we're going to have 1,000 people now go to this one secretary, one admin that is set for that organization to go and qualify every one of us to get the document. So you see, there are the challenges we're facing, right? And I think we got to have more discussion afternoon.
Okay. So I want to give John the last question. Hopefully it's quick, and then we'll leave it up to the committee to sort out and solve that problem for everybody.
Thanks, Mark. Just a quick observation on Afshin's mention about using stimulated cells or even stimulated whole blood to create endogenous molecules. Certainly, I use them for parallelism, but it doesn't replace it. It's definitely better than spike dilution linearity. I do use them a lot to create QC samples, though, which is definitely better than spike samples. Caution, don't do your stability on that, because you will find the number of biomarkers where the stability is different.
So with that, I'd like to thank the organizers again for having us, and thank the panel for all of their work.
Okay, everybody, thanks very much. Please assemble back here at 1:30. Lunch is again on your own. We do have some maps for nearby locations for lunch. If you're joining us for our working lunch, however, if we could just kind of congregate here so we can all head upstairs, that would be great. Yes. I have not seen her. Yep. Chris Leptak, Joe Manetsky. Okay. So we need Chris, Joe Manetsky. Oh, we need those guys too. Two or
maybe you had to run to the restroom. So just Joe. The word is so important.
Yeah, I don't want to create too much of a. Yeah, I think that may just be it. Say again? Even these extra inches will help. Yeah. Yeah. Little bit. Good. Outstanding. Yeah. Good. Thank you. Thank you. Just in the beginning.
Okay, welcome back from the break. Hope you all enjoyed your lunch. We're going to go ahead and get started on the next session. So in this session, we're going to focus on strategies to disseminate and ensure uptake of this framework. The discussion will include potential processes by providing feedback, as well as the development of a communication strategy to socialize the framework to the broader community. As a summary of the immediate next steps, I'll quickly run through that. But before that, I'd like to ask the panelists for the next session to come on up and go ahead and get seated. So just as a summary for the immediate next steps for the framework, the working group will be accepting comments through the end of August on this framework. If you don't have access to the framework right now, it will be made. It is currently available on the Duke Margolis website. We have an event page for this particular event, and the actual framework is there. As I mentioned, the working group will be accepting edits to that. So. It would be really helpful, as many of you know, to make sure that as you're commenting and have feedback on this particular framework, to please do so with direct edits into the document. General comments are somewhat helpful, but it makes it really hard for the working group to really distill that into what that means for the actual framework. So direct edits into the framework would be much appreciated. If you do have comments, I would say make them as specific as possible and make sure you indicate exactly what would need to change and where to make that change in the framework. Those kinds of comments and direct edits would be of most help to the working group. You can send all of that back to the email address that we have, which is the margolisevents@duke.edu. Later, there will be a specific email to go directly to the working group, but for now, please use that. Between now and the end of the calendar year, the working group and the writing groups specifically will continue to meet. We'll review and go through all of the comments and edits that are submitted, and we'll make changes as necessary to the actual document. So for this particular session, I think it would be really helpful for our panelists and all of you to give us very specific ideas on two things, really. One is, what additional groups should we reach out to now to make sure that we're getting a broad range of feedback on the particular work group, as well as, once this thing is final, towards the end of the year, as I mentioned, what is the best strategy to make sure that this framework is widely disseminated, and what's the best strategy for encouraging adoption of what's in the framework and using the framework. So I appreciate comments from the panel on that. With that, I will go ahead and turn it over to the panel, and I'll ask each of you to introduce yourselves, although you have multiple times over the last 2 days, and give any thoughts that you have on these specific questions.
So I think I was the one that was supposed to lead us off. Regarding the comments versus edits, my joke when we met during lunch was comments will be ignored, edits will be considered. So certainly as part of the evidentiary criteria framework document from the workshop about a year ago, being part of that writing group, comments are just really challenging, especially for a group effort where you're working on the phone. So when you write a comment saying unclear, what's unclear? What would you like to change? So again, please, as much as you can, provide specific edits, especially for people that have suggested that the guidance is the work white paper is missing certain sections. And then the other comment they made around the dissemination of information, certainly the two goals are, one, for this white paper to be as good as it can be. Certainly the working group has done a tremendous job putting together a first draft, and certainly the discussions over the last 2 days are going to help that draft to be even better. Not everybody was able to attend today. Maybe people didn't even know this meeting existed, but there are groups out there that can have a valuable input into this space because they're end users, if you will. And so to the extent that you know of groups that would find value or would use information in this paper, please let the working group know who those folks are so that we can reach out to them and get feedback on actual comment edits. That would be particularly helpful from academic groups. They're probably the least represented here today, and it's certainly folks that we engage with as part of BQ on a regular basis. Once the guidance is finalized, not the guidance, I keep saying that because that's where I come from. Once the white paper is finalized, we'll probably do something analogous to what we had done with the evidentiary criteria framework, where ultimately, we're going to put it out on the various websites, Duke Margolis, Critical Path, FDA. The various communication teams can put together, an opportunity to do some type of an announcement so that ultimately people know that it's there. From my perspective at the FDA, and certainly as part of the BQ team, we will be working internally to socialize these concepts once they're finalized with the staff that are the end users on our side. So these would be folks that would be on the qualification development efforts, as well as our more general staff that are encountering biomarker development in the IND space. Not that this would be a direct application to that space, but certainly it'd be something for them to think about and it would be relevant. So those are the things that we would be focusing on from an FDA perspective.
So my name is Joe Manetsky. I am the director of the Biomarkers Consortium at FNIH and deputy director of the Research Partnerships Group there. So my reason for being here, and I guess the discussion topic is very germane, actually, to much of what we've been talking about in the consortium over the past year. And not just in terms of the workshop, but in terms of everything that we do in the consortium, in terms of how do we communicate what we do to folks who would actually be able to use it. So and that goes from, we have a really great marker for measuring kidney safety, which we heard about. How do we get that outside of the project team? And, in the past, the Biomarkers Consortium has been going on for 10 years, like CPATH, and it's a in the initial stages of these projects, you worry about getting the projects done, right? Like get the science. And now I think as time goes on and the area has matured, we really have to start thinking about the next step and making sure that this stuff actually gets out and it doesn't take 10 years between the time that you start talking about a really good marker and it's actually used all the time. So the one thing that I did bring, and I thought it would be instructive, at least to have a little bit of a discussion around, was, it's not that one. So somewhere in there.
These are my slides from session 3 yesterday that got lost.
Oh.
So
Here they are. Do you wanna,
Here they are.
Okay.
I'm probably at the end because that's normally where I am. There we go. That was
There
it.
we go.
That was it.
Nope.
No, the next one back. Okay, so a year ago, the FNIH, the FDA, CPATH was involved. There was a lot of people involved. Had a workshop, middle of April, and it was discussed. The whole point was very similar to this, except for maybe a little broader, which was, what do we do? What kind of evidence do you need for to qualify a biomarker, and putting together a framework to place that in context. Many of you I met there. We were there. Yuri and Frank and others were all part of that. And I thought it was instructive because at that workshop there were 200 people in the room. More than half of them were from industry. A large number were from the FDA. And there were a number of others, and so there was a starting base of a large number of people who were actually there and enthused and involved, and just like here, the conversations were just as active, if not more so, at least as active, and so I thought it would be a little bit of a nice case study in a different way of communication of the outcome. And so the workshop was in April. In October, we finally got to okay, let's send this out. This is what we think it is, right? And so we had the workshop just like this. We had the workshop. We had many months of reiterations afterwards to revise the document, because the workshop like this gave something to react to, and we needed to react to it. So in October, it came out. We sent out to an email to the bunch of people who had gone, and more than half of the people we sent it to opened it up and read it. So that's really good. 50% of the people that were in the room, or mostly in the room, good. In December, we were talking about the kind of a little bit more broad view of sending it out to different people. We did an FNIH e-blast, so this is a communication term that I learned over the last year. And that was also placed at the same time as we sent out things from the FDA listserv. We sent out a bunch of things through CPATH. We kind of blanketed the area. And of those, about 20% were opened. So at least from the ones that were sent out from here. So that's not as good, right? I think it's good. It's actually, to be honest, it's more than, it's several percentage points higher than industry standard for sending out emails. But, there it is. At about the same time, we started realizing that, again, the communication was important, and we were maybe falling behind. We completely redid our web pages and our the website for the consortium, and this was part of it. And so in this case, we had, at the same time, we started being able to measure who was going to the web page and who was looking at this. And we had 300 or so web views on that day anyway, and they lasted for a long time, so they were actually there. That's really very high in terms of industry standards. So, I'm thinking, I'm a metrics guy. So the other thing you got to understand is I may not be the analytical biomarker scientist, but I like metrics. So that's like 6 times the norm. And then if we followed that out, you could see it was picked up at another bunch of places, and some of them came from the FDA site as well as others. One of the interesting things that I wanted to point out about this was that we didn't directly talk to the Regulatory Affairs Professional Society, or RAPS, but they picked up the story, and they actually had a pretty nice article on their website, right? So thank you, RAPS. Did a very nice job. And then I also found a reference to the Myotonic Dystrophy Foundation. So those two were picked up by them by doing nothing, essentially, but sending it out through the FDA. I think we can do better at that. All right, I mean, that's 2. We have it on the FDA site, we have it on CPATH, we have it on FNIH. I mean, these were kind of where they started from, but I think we should be working harder at getting it into some of the other sites. The other thing that we did, which is not on here because my communications folks didn't want me to publicize it too much, was we tried things like LinkedIn and Twitter and things like that. And that was, from my point of view, pretty bad. It really did not go well. And we didn't get a lot of retweets. We didn't get a lot of...
What was the hashtag that you used?
It was... So, it might not have been as specific as the one that's being used today. But it, but particularly like LinkedIn, I was a little surprised we didn't get a lot of reposts and things like that. And I was just curious, like, this is something that we all care about, I would assume a lot. Just for my own edification, could you raise the hands if I sent you a LinkedIn note saying here's something? How many of you would actually repost that? Yeah. So maybe 2. So that's about right. Yeah. There was how many actually would know how to repost? No, don't say. I had to be taught. Yeah. So that was, I actually had to go back and find out how to do that. So at any rate, that was something that, if we try to do that again, that we would have to approach that a different way. So I think from this point of view, I look at this and I say, we're doing, maybe we should be doing better. We should be taking this on as a much stronger and much bigger part of what our daily life is. And this communication process actually depends really on everyone. And I think in the past, we've all had the situation of, well, FNIH will handle the communications, right? Well, that's their job, that's their thing. But I think what we really need to get to a point where it's we all do this. This is everyone takes a very active role in reposting and in making sure that people are out there listening, because otherwise it's not consensus. It's not consensus until it's absolutely clear that the groups who were involved in it all buy in and are all promoting. And so that may sound a little bit like I'm trying to convert you, but I think you need to. We need to do this. So that was my perspective, and I'll pass it off to Lisa.
Back. That's right. I had a quick glimpse of Lisa's. I can do it for you.
Yeah, you can do it. Sure. I can't hold two things and talk at the same time.
I'll help you.
So as was already noted, there probably are not that many people representing academia in the audience today. If you consider yourself primarily an academician, raise your hand. Okay, so just a handful. So part of the reason I'm here is that the community I deal with are the academicians. And so I wanted to kind of give you a perspective for where they see this whole biomarker qualification effort and what role they can play and how we could encourage them to do things in a way that would be more helpful to some of these big qualification efforts. And I guess the first thing I would observe is that I see many people in academia use the term qualification, they use the term validation, they use other terms related to biomarkers like surrogate. They have no clue what they're talking about, okay? It's all sort of the same mush of things to them, okay? So I think that's an important role that we can play to make them understand what is the essence of a qualification program and how is that different from validation in the many different forms of validation we have, and how is that different from getting an FDA device cleared or approved. So it's all, I'll tell you, it's very, very muddy in the minds of many of the people in academia. Next. But we should point out that the people in academia still are making very important contributions to drug discovery and identification of biomarkers that might help us to better use those drugs, target them to the right patients. And one thing we have found to be a very big challenge is that the integration of laboratory and clinical sciences is suboptimal, to put it lightly, in many academic institutions. There are people who are doing the clinical trials, there are people who are doing the biomarker laboratory work. You might have a clinical laboratory in a big institution, and those three parties do not necessarily talk well to one another. And so we have found, as we have gotten more into targeted therapies, for which there might be a biomarker used, say, in an early phase 1/2 kind of trial, where they're going to enrich on a biomarker for entry into the trial, we began to realize that some of these biomarker assays had like no clinical validity assessment ever done on them. And in fact, some of these maybe had never even been performed. So we, a few years ago, instituted a process called Biomarker Review Committee, where we had a little checklist and said, tell us, first of all, how you do your assay, and tell us a couple of basic pieces of information, like relating to analytical performance. Tell us what reproducibility studies you've done. Have you assessed the sensitivity or specificity, reproducibility across different readers, different labs? Tell us what you've done. We're not expecting you will have done all of these things, but we were sort of hoping they would have done a couple of them. And so we started using these forms, and were, I have to say, horrified at the answers we were getting back in these forms. And I have some funny examples that I've given to some of you. I won't repeat all of those because I'm not trying to make fun of people, but it just really told us that people do not even understand some of the basic terms. So what was happening is your clinical, often a junior clinical person, was put in charge of an early phase trial, and they were told, we got to put this biomarker in, and they'd try and go find someone in a lab who was willing to talk to them about doing the assay. But that line of communication and getting the time of that person who had the experience in assay development and assay analytical performance evaluation was really tough for the clinical people. So by instituting this new review, we were kind of forcing a marriage between them. And it wasn't always a happy marriage. And so I think that that we're continuing to do more education to say that you really do need to have this conversation, and we don't want you to do your early phase trial, enriching on a biomarker that nobody else will ever be able to reproduce. And we're not asking a lot here, and we're not, we've heard a lot about the nice full analytical validation process. We're asking for really basic stuff, like, can you do 10 samples, split them in half, and run them on 2 different days, and what's the concordance? I mean, this is the level we're starting at. And so it's really basic stuff, and we need the help of all of you to get the word out so people understand what are some really minimal criteria you need to do to be sure that it's worth even doing this biomarker. And we're in our early phase trials, we're talking about patients with advanced disease. You're doing a core needle biopsy in most cases to get that sample. It's unethical to waste those samples, put the patients through that, to run biomarker assays that give you crap results. So, I think we really need to take very seriously what we're doing here. Somebody, I think it was the gentleman here, had mentioned about the attention we've put on rigor and reproducibility in research. And NIH has been put on the hot seat and said, what are you doing to make sure that all of these grants that you are funding using taxpayers' money is resulting in research that is usable and reproducible? So about a year ago, I guess it was, after many deliberations amongst the leadership of NIH, they actually put out some guidelines on the grants website saying what they're going to be doing and how they're altering their grants review process to hopefully improve the reproducibility of the research that we are paying for. And that's what you see here. This is pulled right off of the grants website. And just a couple of things that are of particular relevance to what we've been talking about over the last 2 days is, first of all, and this is another hat I wear, I'm very much into reporting guidelines for research. So there are many papers you can pick up, and even good journals, and you read through the paper in the methods section, and you still are kind of scratching your heads like, well, how did they really do this assay? And how do I know that that antibody they used in their assay was in any kind of a decent antibody? And so we've been giving these instructions to our grant reviewers that they need to really look carefully at the background information provided in the grant to assess how good the evidence really is. And people have different levels of grantsmanship skill, and some can write volumes of stuff that sounds beautiful, but when you sit down and actually say, what did they say, there's not a lot there. So we're just being a little more prescriptive about the kinds of things we want to see. And so some more specific things are rigorous experimental design. So if you're doing a study, early drug study, maybe even a preclinical study, how did you decide you were going to do 5 mice in each group? And tell us more about how you picked some of the reagents for your assays. Did you validate them? If it's a cell line, did you validate it? If it's an antibody, did you actually validate it before you used it in your experiment? Because it's the old garbage in, garbage out. If you haven't made sure you're working with good basic materials and you haven't sized your study to actually be able to answer a question or designed it in a way that avoids bias so that you can answer a question, then we don't want to proceed with this. But often that information is not given. So we're, again, having more information we're requesting to hopefully do a better and more fair evaluation of the research proposal. And I think, do I have one more slide, or was that it? Okay. So the goals of all of this effort, and I think what we need to accomplish here, is to raise awareness of the importance and the impact of assay analytical performance. And I get to see some of the crash and burns that you guys never get to see. I mean, I've seen the grant where they had an assay and somebody switched a reagent halfway through, and the investigators contact us and say, well, we just did a plot over time of our assay results, and it's a totally different assay as of January 2015. And I've actually seen people give back their grant money saying, I'm just throwing in the towel. Okay, so I get to see that stuff that you guys never get to see. We do need to educate all the stakeholders. There really is a big need. And even in academic institutions, they have terrific clinical labs, terrific research labs, but the research people don't necessarily know how to put an assay through all the paces that you guys have been talking about. And even if they did know, they frankly don't want to spend their time doing that. People who are running the clinical labs know that they have to meet clear requirements, and they have more attention paid to that. But it's often the research labs that our academic clinical people are working with who are doing our early phase trials. So we need to have better communication going there. And so we do need to incentivize this because it takes time and it takes resources. And one of some of the feedback we get when we ask for our biomarker checklist to be filled out is like, I don't know where I'm going to get those specimens. Who's going to pay for me to do these assays? And it's a big issue. And so we, as a funding institution, also have to think about how we can not only incentivize, but also provide resources so that people can do some of this important work. And finally, I think our goal needs to be to share this biomarker data that we're generating. Now, data are only going to be valuable if, number one, they're any good, and number two, you need to have accompanying those data adequate documentation of the assays, the reagents, et cetera, pre-analytical factors, so that we can judge if the data are any good. And any of you who have been involved in these biomarker qualification projects know, I mean, you're often going out searching for any data you can find that might be useful for your qualification effort. And it would be a real shame to not make use of these data that are being generated in academia and other places to really bring it all together. And even if we see differences in the data, we learn from the differences. But observing differences doesn't do us any good if we don't have other variables recorded with it to understand what might be responsible for those differences. So it's really a matter of trying to do our science in a more rigorous way, documenting in a better way what we've done, what we've used, and hopefully we're all going to benefit from that, not just academia, but the entire community. Thank you.
Great, thank you.
And Lisa, I quote you often because as part of conversations we've had in the past, when the outside party says, look, you're trying to tell me what to do, Lisa says, no, I'm not trying to tell you what to do. I'm trying to give you the tools to tell us what you did.
No, we get that response a lot with our reporting guidelines. Say, I don't like reporting guidelines. I don't want to have to follow what somebody else is telling me to do. And I give exactly the response that Chris said. You can do whatever you want to do, and people doing different things is actually a good thing because we can learn from it. But if you didn't tell me how you did it, it's not useful to me.
Great, thank you. Martha?
I'm Martha Brumfield, CEO of Critical Path Institute, and really appreciate all the comments Lisa made because we deal with a lot of educational aspects within CPath as well. So will the slides come up?
I bet you want that slide to pop up.
It would help,
you
but
figured
if not,
this out,
that's
didn't
okay.
you? What's going on? Next slide, please.
Well, not to waste time, I'll go ahead and start talking.
Yeah. Is that the
Back
first
up.
one?
No, back up one.
Okay.
There you go.
I did hit it a number of times.
It's okay. Go forward one.
Okay.
Sorry. One thing that we are poised to do because of the nature of our work is to help get the word out about the broader framework document and also this white paper once it's ready to be further disseminated. We run a lot of consortia, and I listed in the first bullet just the number of biomarker programs that we're working on as of today. And that's not the total number of biomarkers, but the number of programs. So it's Alzheimer's disease, Parkinson's disease, Duchenne, the safety biomarkers, tuberculosis, and on and on. And we have found whenever we start a new consortium, and typically they're constituted of scientists from academia, scientists from the regulated pharmaceutical industry, occasionally from device companies, government agencies, FDA and EMA also very much support through a liaison role. But we probably spend the first year educating that whole group of people about what qualification is and what we're going to need to do to get a biomarker across the line. So it's educating them about the importance of clinical validation, but certainly with the assay validation. And I would say we have found the amount of time it takes to get a biomarker across that goal line really hinges on the quality of that data and how much validation has been done up front. Just in the last month, a new consortium we've started, we've been trying to get access to a data set for which we were told it's perfect, it's got all the biomarkers you need, it's longitudinal data, and we get the data and it's not even half of what the individual said was there. So I don't mean to imply people are trying to misrepresent information. I think there's a fundamental lack of understanding of what kind of data one needs to make a regulatory decision as opposed to publish in a medical journal. So I think we all have a lot more work to do. And as others have said, we need your help to get the word out to your communities. We certainly will work with our communities. We will disseminate information throughout all of our consortia, and even to groups that we choose not to work with. Multiple times in the course of a year, an investigator will approach one of us and claim, I've got the best biomarker in Alzheimer's disease you've ever seen. I've done it in 10 rats. How do I qualify this? So this is where the conversation becomes, is it regulatory ready? A term John Michael used earlier. It's a term we use a lot because we all know there is a very long journey from discovery in the laboratory, looking in animals, and then translating that into humans. So documents like what the working group has prepared and we've been discussing the last couple of days are really going to make a difference in being able to get biomarkers and hopefully other kinds of drug development tools and novel methodologies out there that ultimately will de-risk decision-making, both for those developing the drugs, but also for the regulators that need to ultimately make a benefit-risk decision. So I'll transition to the next slide. I just wanted to point out something. I know Chavre spoke to this yesterday, and I think Chris has mentioned it, but it came up in the discussion earlier today about the importance of having real examples out there where others can learn from. So under 21st Century Cures, with this new provision for transparency, this is going to happen much faster than it has in the past. With the biomarkers that have been qualified, the information is made available at the end of the 6 or 10-year process. But now information is going to be made available much earlier in the process. Now, I can't speak to exactly how much, but for example, at step 2, where a qualification plan would be submitted to the agency, it's going to articulate what does that analytical validation look like. So there's going to be at least reference to what your goals are, and that's going to be informative. And it's certainly my hope that as more of this information becomes public, we as a community are going to learn and we'll get better at this so that we can move things through the cycle much faster. So it provides an opportunity to learn, and another of my personal hopes is it provides an opportunity for us to sort of combine forces and work more collaboratively. Because the last thing that the agency needs, or any of us, are 5 different groups working on the same biomarker and coming up with 5 different ways to approach that biomarker. So we really need to think about enhancing our collaboration. I think there's one more slide, and I'm just going to summarize what Greg started with, is that for now, we want comments, particularly edits, very specific edits, sent to margolisevents@duke.edu. We'll be changing this to another email address in short time, but you don't have to worry about that. If you send them here, they'll get forwarded. I'd like your input on, are there other professional societies that we should be circulating the document to now, or other networks that some of us may have where we can reach a broader group of folks that have a vested interest in what we're discussing here. We needed to set a time frame, so we're going to say by end of August, we would like comments and edits on this white paper. The writing group, you've done a ton of work, but you got a lot more to do, I'm sure you know. So they will decide at what pace they're going to be able to work, but they will review the comments, and then I think it will be up to them to decide how best to address comments and get another version or a final version or whatever it's going to be. So too soon to tell you exactly what that will look like. But certainly we want to make sure that everyone that's participated in this workshop, both here in person and those who are online, that this comes back to you so you can see how it's been refreshed and incorporates the amazing discussion that we've all had here today. So I think with that, I will stop.
Okay, great. Thanks for all of the comments. And I just want to reiterate the point that although that email might be changing going forward, the actual website on the Duke Margolis site that captures the video and discussion from the last 2 days will stay up for, I think we said it, not quite eternity, but for a really long time, much longer than will probably be needed. But
as long as Duke is in existence, the website will exist.
We decided. So that website will stay up. The draft document will will be on there with and accepting direct edits and very specific comments through the end of August. So I do want to ask, we have some time left on this, and so to the group, questions about which groups or networks should we be reaching out to and how to drive adoption. But one just quick question to the panel, if you can just answer one or two of you in like 1 minute, is that Joe, despite all of your efforts on Twitter and LinkedIn, you've shown that it's really hard to get people to look at things and to sort of actually adopt things. And Lisa, you mentioned just how hard it is to get academic researchers to use the tools that actually can help them be more transparent. But to Martha's point, that this is really about de-risking decision making in drug development. So what can be done to help encourage adoption of this framework in like a few words only?
Make it readable for the academic biologist.
Yeah. Okay.
Can I ask just a really simple, can you make it available in Word format so that everybody does it in change edit mode? That's going to make your life so much easier than sending it as PDF.
Okay. We'll make sure it's in that mode. Yeah.
So the one thing I would add is to everyone here has to be involved.
Yeah.
So
not just the individuals, but the organizations. And,
yes.
Yeah.
You have to be actively reaching out and pushing the envelopes of maybe some scientists' social skills.
Just really quick, procedurally, I would strongly recommend that you include line numbers in the draft version that you put out. I'd hesitate about putting track changes in Word depending on the number of comments you anticipate. It would be really hard to merge, but line numbers as a PDF and make people comment on what line number. This works in standard organizations, other places. Just practical recommendation.
So with regard to your question about how do we disseminate the information, so the one thing you notice here is there are a number of members from the American Association of Pharmaceutical Scientists. Many of them are technical experts, so we can definitely reassure you that we will take the message to the technical experts. But what we do need to have in the mix are the academic and the medical experts, right? We cannot ignore them. So my question is, and it's just a suggestion, like Michael J. Fox Foundation, which really funds a lot of biomarker initiatives and efforts. So is there a possibility, like we have members from the Huntington's Disease Foundation in the audience, but can we take it and provide it to them when it's in near final form to say, whenever you are providing a grant, can you provide this as a guidance to all those people who are receiving grants? So similarly to the Alzheimer's efforts, I've worked in so many different consortia for Alzheimer's, and fortunately for us, we've had very strong clinical partners. Nevertheless, it's taken such a long time for any of these CSF biomarkers to see the light of day. And the issues are all to do with technologies and how people have approached assay validation and clinical validation. So I think that is something for us to consider with regard to how do you disseminate the information. Take it to the American Society of Neurology. Just ask them to post it on their website for anybody who's embarking on research activities. Would you consider, following these procedures because it'll simplify your life later?
It's not a secret society with a special handshake. So once this comes public, especially once it's near the final form, please share frequently and often. Put it in, your holiday gift certificates, whatever you want to do. But certainly get the word out. And all of those groups that do work with especially patient advocacy, many of which raise funds for biomarker development, it's important for them to know what, not only that they want to develop the biomarker, but then what they're facing so that they have realistic expectations.
So, and when I'm talking about, pushing out the work that we're doing here at this workshop, we see a number of biomarker projects each year, and I have to remind all of those teams every time I talk to them almost to go to the best toolbox and use the correct name for the biomarker. Is it a response marker? Is it a prognostic? Because everybody has these things in their head and they're all different. So it's not just, it's a package, right? It's this validation, it'll be the statistics, it'll be the framework that's already out there, it's the best glossary. All of that has to be, reinforced by the groups that are here. So yeah, put it in your Christmas list.
Well, if I could add, I'll certainly make the commitment on the behalf of CPAC that we will push this out to members of all our consortia, and Michael J. Fox is one, as is CHDI. So we can't tell them they should use it, but we can tell them this represents the best thinking of this collective group of experts, and that we want their comments, and they should be aware this is the direction we're moving. So we can certainly do that.
So you guys are speaking my language right now, because this is why I'm arguing for having some transition for this document, making it accessible. And I'd love to know now what you guys think over there, because this doesn't sound like what you're wanting this document to be. But I don't want it to be all validation all the time. But my whole thought process was the fact that I do work with academics. I do work with kind of these small institutes. I do work with all sorts of people who aren't experts. And I don't want it to be all validation all the time, but I do need some transition for the people who aren't big pharma or big biotech to use this document and figure out how it applies to them, right? And it can't be the very end, an FDA submission. It can't. I know it has to be somewhere before that where they jump into the document and they can access it.
Mark, if I could...
Question over here.
I think it was unanimously, we want you to stand in line, Mark. So, a philosophical question, guys. I don't like to get into this philosophical discussion. I think, Lisa, this is more toward your comment. I'd like to get your input and the rest of the panels. Look, we all know the basic research science, the brain, the power behind a lot of the drug development comes from academia. There are pros and cons of what academia can do. Let me give you some examples. You pointed to something. I inherit as a CRO clinical trial samples that have gone through 9 freestyle cycles because the lab kept running. I have to do 12 freestyle. The samples are missing. I send academic lab water, and they report 1 mg per ml of ghrelin in there for me, okay? I go in there. I was a graduate student myself, and we know you get that pipette, and you basically calibrate it when it comes, and 10 years later, after 5 graduate students drop it on the floor, you throw that in trash. That was the level of calibration of the pipette, right? So I want to make sure that, again, we come back to planet Earth and not in a utopian world. This document, right, we want to get valuable input in order to make a decision as what to do and move forward. So the stakeholders, I get it. This is, again, if you are, again, suggesting this paper is we're going to send it out, and we're going to go ahead and through this paper, and even if it's finalized, we're going to train our academic colleagues, okay, on what is the difference between how to develop a biomarker, what is the difference between precision and accuracy. I don't think that's the way to go, okay? And I have some ideas. If you want, we can talk over a beer of what NIH need to do and hold them accountable for some training program, training files, and then give them grants, okay? But so for this one, though, again, and this is, again, we're talking about bioanalytical assay validation for clinical biomarker qualification. So to me, no disrespect to any of the academic lab over the phone, this needs to be done. We have to find, again, some of these consortiums. This got to go to every pharma, biotech, CRO in the world as much as we can say and get their input. For somebody who still does not know the difference between precision and accuracy, even if they're expert in oncology and Alzheimer's and whatever else, I'm not sure what value it will add. Okay, so that's just my comment.
I still think you and I are having a disconnect about what we mean when we say biomarker qualification. Okay. Because we have a lot of academics that engage with us for which we can qualify a context of use if they have the data. And it's not to the extent that there's a marketable assay or anything to that caliber. And that's okay for us depending on the context of use.
Right, but what that means here is that 99% of FDA's time is spent on correcting and teaching, again, some of these laboratories, right, versus using those resources for the biomarker work that comes out of the pharma and biotech that is a lot more tidied up. I'm not saying that, no?
I don't disagree. What I'm saying is if we're going to hold early phase, non-risk, no change to patients in the clinical trial, and we're going to hold them to a level of standard clinical validation that would be the same as a CDRH clearance or approval, we will never learn anything in the science space because it'll never get incorporated in clinical trials where we can collect data to learn more. Okay. That's my point.
Points well taken, but again,
Let me just.
go ahead,
we
please.
do have
Yeah.
like 5 minutes left,
No,
so important points. But let's.
This is another 4 or 5 beers when you have a conversation. Go ahead.
I
over here.
just have a a very good, very simple point that I think that the hashtag help us promote biomarkers for biomarker qualification would be a good one. That was a joke, okay?
Yeah.
So now
I really appreciate it too.
I saw your face. Like, see me. So I've got to qualify. No. The real point is with all these kind of a communication tools, right? We left out one. And the one is the peer-reviewed publications, because we need to get it in the literature. We had issues getting our one in, but we need to really concentrate on that because, as you said, who posts something on the LinkedIn? And I said, I can post something on LinkedIn. So that's, you have the audience. If that's your metrics, okay, yeah, it's really, really bad. But who read a paper? Yeah, everybody, right? So we need to get it in the literature, and that's it. I'm resting my case.
There is a comment behind you. Why don't we?
Can I just say
is it
the end
just on
goal
this?
of the
Okay.
white paper was to publish it?
Okay. Next, please.
Hi, I'm Jen Madsen. I'm with Arnold and Porter K. Scholler, a law firm. I'm not a lawyer, but I was thinking about this a little bit just in the context of having done a lot of work in the clinical lab and pathology world that, you've got sort of two totally different populations of people. The kids doing research in graduate school, in the research lab, and the kids that are going to get the MT technologist certification from the ASCP are not the same people. And they don't have the same training. And so I think that some of that is things where, in order to kind of get the research lab and the clinical lab to sort of understand each other better, you've got to sort of start with the thought that we're not even talking about the same populations of, college students. But then, further from that, I think, when you look at the academic setting and sort of where papers need to be published, the archives of laboratory and pathology, engage with the Association of Pathology Chairs, which represents all the chairs of all the departments in the country. And I think that, by getting things sort of better coordinated between, oncology and pathology and what have you, that would actually it would probably help a good bit, actually, because I just keep finding that drug folks and device folks and diagnostics folks and lab folks just don't all speak the same language, and that seems to come up at every one of these meetings that I attend.
Okay. All right. Thank you. So another comment?
Hi, I'm Cindy Lancaster. I work in regulatory affairs at AstraZeneca. I've worked with the Critical Path Institute on some projects in the past and Transcelerate. And so I'd like to offer up, I'm not as technically background as some of the folks in here that do validation work, but I've spent a lot of time working with development folks who don't necessarily know about device and biomarker qualifications. And part of what you could probably help them with is having a community of practice to kind of implement this once you get it published. I certainly agree with all the comments that have been made about things that would be helpful, but a lot of times people don't know what they don't know until they need it. And so the other thing you could do is publish a list of low-hanging fruit, because I think FDA said that they've had, 2 examples of prognostic biomarkers. I read the BEST paper, got a little bit of education. My background, I cut my teeth on hypertension and heart failure studies a long time ago, and so, I saw blood pressure there listed. Maybe people need just a little bit of guidance about, here's a list of low-hanging fruit. If I'm working in this area, who do I contact to get a little bit of education? Now, it's a lot of work to have a community of practice, but if you're going to publish, and you need to do something to help people implement, because again, I'll go back to people don't necessarily know what they don't know until they're in over their heads. And if you want basic data from the scientists, they have to know how to get it to you, and they need to know that what you're talking about is they have. So that would be my thoughts.
Great, thank you.
Can I comment
Responses?
a little bit, on the issue of will the paper that's already been drafted serve the academic community? And I agree, I don't think your average academic, unless they're really in tune to biomarker qualification, is going to sit down and digest that paper word for word. But what we can do is put it out there as this is a destination where some of the biomarkers you're working on could wind up. And if we appropriately reference things, have a lot of references to more basic papers that explain some of the fundamentals, and there are such ones. I could tell you some for like the statistical concepts in analytical validation, that they might at least find that valuable. So, write it in such a way so that they get the concept. They can say, well, okay, I'm not there yet, but oh, gee, look at all these things I'm going to have to worry about, and here are a couple of references that might give me the background that I'm currently missing to understand this situation better.
So just a 30-second response. There are great white papers out there that they will do a better job serving that audience for exactly the purpose you mentioned. If an academic lab doing biomarker work and they're not aware of Gene Lee paper, we got a problem. That lab got to go start from scratch and start moving forward. I get it. I don't agree with it.
I've not read Gene Lee's paper, but I think one of the biomarker qualification advantages is that you can put it in that whole framework of understanding the big picture. And so it's not the same as reading a paper. And again, I don't know what Gene's paper says, but if it's focusing primarily on the analytical validation part, it may be extremely useful for that. But is it putting in this bigger framework of understanding your clinical context and all those other things that you need to be thinking about simultaneously to gather the right evidence?
Great. So that about wraps up this session. But I know the very specific question that we had was ideas on additional groups to sort of reach out to and bring in comments. So I think throughout some of the discussions, I heard quite a few sort of societies like American Society of Neurology, Michael J. Fox Foundation, so societies and patient groups, et cetera. I also heard industry, reaching out more to industry and getting feedback from those groups too, which I think is a good...
Howard Hughes.
Howard Hughes, okay.
I think Howard Hughes would be a great institute for the clinical community that are interested in research, especially since many of them become academics.
Yeah, so the other is pharma. So
right,
the Pharmaceutical Manufacturers Association, and bio. Bio. DIA.
DIA has broad reach.
Yeah. And there are very specific people in that area.
Okay. There we go. Okay, now we're flowing.
And
NCATS is a good
Precision Medicine Initiative.
Okay, that's right. Okay.
GCC, EBF, JBF.
Okay, great. Also, line items, line numbers instead of track changes is really going to be helpful. We'll make sure that, as I mentioned, that the web address and the email is broadly communicated too. So with that, I'd like to thank the panelists for the great discussion and all of you for your comments. We'll move now directly into our next and final session. Thank you. Does Mark need this? Okay.
I'll use this one for now, unless you want to stand. I'm thinking maybe you could sit here and I'll just.
I'm actually good with standing.
Okay.
Yeah, I'm good with standing. I don't mind. We can kind of share.
All right. If all of our panelists for the final session would come on up, we're gonna go right into it. So I want to add my thanks to. Are we good here? Good here. Can you all hear me? Okay. All right. I want to add my thanks to Greg's, to all of you who have contributed to the efforts over the last 2 days, and of course everything that went into the white paper in preparation before then. It's great to have such a distinguished group, and it's great to see people so committed to sticking with this effort through 2 full days of meetings. For this session, Shavri and I are going to moderate a review of some of the major themes and topics that came up over the course of the last couple of days, have a chance to talk about some of the major takeaways, prioritize some of the next steps towards the completion of the white paper and how it fits into this very important pathway towards better evidence related to biomarkers. So we're going to try to make this a back and forth session. Our panelists had a chance to reflect over the lunch break on some of the themes that they thought were most prominent, and so we're going to start with that, but we very much want the participation from all of you and people who are still with us online. We've had a lot of participants online over the last couple of days. So Shavri, maybe I'll turn to you first for any opening comments, and we're going to pull up a slide in just a second too that describes some of these main takeaway themes.
Sure, thank you. And I wanted to echo our thanks to everyone, especially the writing group who's done a tremendous job of making this something tangible for us to react to. I do want to make one clarification. I've heard we use the word guidance and we kind of throw different words around. So there's big G guidance, which is when us at FDA put things out, we think of big G guidance. There's little g guidance, and we kind of confuse those terms. This white paper that we're developing is not big G guidance, okay? I think it is an important component of a variety of documents that we will take a look at and think about as we develop big G guidance. So I want to make sure people are clear about that and we're not confusing terminologies. But I cannot understate enough how critical it is that the timing on this is so important as it informs where we're going to need to go. Biomarker qualification is a big deal at this point, not only because of 21st Century Cures legislation, but also because of the upcoming hopeful PDUFA 6 reauthorization, where it clearly speaks to the need to develop guidance for evidentiary considerations for qualification. So I'd like us to do introductions maybe, and then we'll go from there.
Yeah, and if you all want to go ahead and do the introductions yourselves. Yeah.
Sure. So my name's Russ Grant, LabCorp. For those of you, this is the face that you've been hearing. Sorry about that. I've been fortunate enough to be at the other side of this conversation in diagnostic medicine. So 15 years actually validating, deploying, transferring FDA approved and also LDTs in the direct care of patients. So obviously I'm coming from a different angle back to this interesting conundrum. I'm personally thrilled with the positive feedback, and I'm very, very thrilled with the engagement that we've had over the last 2 days. But I'm also a little perturbed, and so I'm going to use the words of, you don't know what's coming yet, and you're still chuckling. This is good, right? So, you know, the galaxy's greatest philosopher, Master Yoda, and he said, fear leads to anger, anger leads to hate, and hate leads to suffering. Fear is the path to the dark side.
Always with you, bias it is.
You got that right.
Now, was that rehearsed? Yes. All right.
So what I want to communicate is that there should not be. You were, Sujane, you were worthy not being there. This. There is lots of support and lots of reaching back across a boundary, and I would just recommend that if you know clinical chemists, we are talking a different language to some in the room, but a very common language in that context. So feel free to talk to a good old clinical chemist or even a young one, because they're a chatty bunch, and it's been a pleasure so far.
My name is Yoda, and I'm short and green and have large ears. Steve Piccoli. I have a long history in clinical chemistry, diagnostic and pharmaceutical industries, and actually even a stint at the FDA on a medical devices panel, so I have seen all sides of this argument that exist. I've spent much of my career designing tests to do very specific things in the diagnostic or drug development continuum.
No problem. My name is Sujian Wang. I'm at the FDA for more than 20 years. Prior to joining the FDA, I was at the Medical Genetic Institute, so I was doing a lot of pharmacogenomic stuff. Coming to the FDA, I then do a lot more on the clinical trial, so I see both sides from the academia, semi-academia to regulatory, but not industry. But through this discussion today, I finally realized that when I see a phase 1 protocol, and I will see a statement said, oh, the assay is fully validated, but there's no number or anything like that. But we don't question further, that was a phase 1. In terms of the biomarker qualification program, I joined the group, start from the very beginning until now. And I am pretty happy that the statistical team, although I heard a lot of statistician comments during the meeting, but I'm pretty sure our stat team really contribute a whole lot to get the 3 clinical biomarker being qualified. So for that, I think I finally hear more statisticians' participation when we receive any biomarker qualification submissions. For today's takeaway, for me, because this is for biomarker qualification rather than for drug approval, I thought I heard from the various parties, and I come down to this one takeaway message. I hear that the analytical validation is a continuous process, so at the high level, I think it may be good to have an analytical validation roadmap at the high level. And there, you will then be honest with what you have done from a regulatory ready assay, design your assay with a biological variability very well characterized. So give all those information to us in a way that is representing the major information or major result that you want to share with us, rather than just say it's fully validated. So we see those numbers and we know what's going on, and we have confidence in the assay itself, so that we have confidence of the biomarker that's going to be measured.
Am I going to limit myself to introduction or comments to?
I think introduction and brief comment if you want, but we're going to have plenty of time for that, and any impressions you'd like to do.
Okay. No, I'm not good at that. Steve, you're it. Okay, now just to give me my background, after my postdoc work at UCLA, I joined a diagnostics company, and then I was in a biotech company, then in a non-profit organization before joining FDA. So I have seen it all except the pharmaceutical industry, I think, but we have indirectly dealt with that. So that's my background. And one thing I wanted to clarify is that we have never denied qualification of a particular biomarker submission because the assay was bad when they came in. We would say, hey, you need to do this, this, and this to improve the assay. So it's not such a deal breaker. If something is missing, we'll let you know. Just hope that you come to us with that early, and with the new process, you will have to come to us early with all the analytical validation. And I echo what Chris said about how the document should be more readable, and we heard from others also in the audience. And we do have about 70% of our submissions come from consortia. We also have submissions from academia, CROs, disease foundations, and diagnostics companies too. So there is a wide variety of people who send in these submissions. So it'll be great if the document is something that's readable by any biomarker developer.
Great, thanks. Thanks, Shashi.
Good afternoon, everyone. My name is John Cadival, and I'm a team lead with the Office of Study Integrity and Surveillance. I am one of the co-authors of the 2013 revised draft guidance for bioanalytical method validation. A lot of familiar faces here from Crystal Cities and WIRBs and other meetings, of course. OSIS, as we would call our office under the acronym, we are the custodians for bioanalytical and clinical inspections for bioequivalence, bioavailability, immunogenicity, ADA assays, as well as biomarker assays. So our touch point is very different from a lot of the discussion that you've heard over the last 48 hours. It hasn't been quite 48 hours, unless you include, yeah, it seems like it probably. So it's a little later in that stage. So by the time I and my colleagues see a biomarker assay for an inspection, it's very critical for me to be here to understand the series of events, to connect from qualification to the stage where it might be serving in a phase 3 safety and efficacy study. So that's why the dialogue here has been very important to me to understand that. And also, and I'm pretty sure I've read about 3,000 of your comments for the bioanalytical method validation guidance. I'm not exaggerating, because we had 6,000. So I got half of them. So the important takeaway is we saw the evolution of the need for biomarker attention when it comes to analytical assay validation. But for the last 2 days, it was important to see how it fits in qualification, and also, not that we have to talk about it right now, but down the road, how's it going to fit with further drug development under IND, NDA stages. Thank you.
Hi, John Michael Sauer, Critical Path Institute. I'm going to use my time to really thank the team. I mean, I think you've seen a demonstration of what this team has been like working with them for a year. It's actually been a lot of fun putting this paper together, and I really look forward to finishing it up with everyone's comments.
Edits, not comments, right?
That's right, edits, direct edits into the document. So really look forward to that. I mean, we still have more work to do. I mean, we created the framework. That was a great step forward for qualification in general. I think here we can come out with a bioanalytical framework, a validation framework. I mean, there's more to do around qualification. I mean, we're going to talk about in the future about imaging biomarkers and what's the validation required there. We still have a statistical working group that's going on. Sue Jane plays an important role in that. And there's a little bit more to do. And once we get this all established, I think qualifying biomarkers is going to be relatively easy, because we're trying to find that consensus now across industry, across academia, across FDA and other regulatory agencies on how to move this forward.
John Michael, do you want to take the first 2 bullets on the slide and then
I should say we've got, because of the work of this group in putting together key themes and takeaways, we've got 4 slides that we're going to go through. We're going to do this as kind of a back and forth between different people in the panel, and then we are going to try to leave plenty of time for comments and questions from all of you. I think we'd like to go through all of the main points, but if there is a burning question or comment or clarification that you'd like as we go along, please let us know, and we'll pause for that.
Perfect. I mean, the first 2 are basically word for word what I discussed this morning, right? Around the fact that there have to be some core expectations regardless of the biomarker. And I think we all agree with that. I don't know. You know, there might be a couple lingering thoughts out there for qualification of biomarkers in the assay validation. And then I think the other piece that we really aligned around was the fact that that context of use, the behavior of the biomarker, the patient population is going to then drive what additional validation expectations is going to be associated with that final validation. The bottom one we modified during the discussion, right?
Wes
and what we
wants to take that one.
Oh, I'm sorry.
He wants to.
Oh no, go ahead.
This is what we mean by back and forth.
This is my Rust to Rust moment. So again, hello, brother. Exploratory biomarkers are outside of the scope of the white paper, clearly listed on page in the intro section. And the however statement, we just wanted to just refine what we mean in the however statement. So we've added here, just for a bit more clarity, the principles of the white paper may be applied at the discretion of the investigator in exploratory situations. Let us at least just briefly explore the idea there. We had a beautiful word earlier, which was de-risking. So a little bit more work up front. The principles applied maybe earlier. I know we've talked about creep. I get it. And Russ, honestly, when I asked you, do you do a little bit more work earlier to make an informed kill decision? I graciously accept that you said yes. And that is not a bad thing, because that's de-risking with a little bit more evidentiary data. So we're trying to leave the latitude in there, even though the flavor is really pointing to that phase 2, phase 3 pivot. We are trying to generate a principled document and process that doesn't apply, but at least is something to lean up against, or at least a framework, or even a milestone. That's where Oz is. You're on a yellow brick road. Are you going that way, or are you going to be taken by flying monkeys? Up to you. But that's where you've got to go. So it's really to try and give some feel for a waypoint or a marker post, and that's the intent. So thank you for your honest answers, Russ.
And before we go on to the next slide, any other comments on these themes from the other panelists?
I just wanted to ask the crowd.
And
Shakshi,
microphone.
Mike.
Sorry, I just wanted to ask if there was a lot of discussion about whether this should move to the front of the paragraph or the back of the paragraph. If we bolded it, would that make everybody happy, irrespective of where it lives? Okay.
No strong views. Okay.
Who wants to take this one?
Whose turn?
Steve, you want to?
Key themes and takeaways. Biomarker assay validation in the biomarker qualification space cannot be done in isolation. It requires input from other scientific disciplines. The clinicians who want this test to be done to define the clinical statement that they need to be tested, biologists to allow us to examine the underlying systems biology of the patient, and statisticians to let us know when we have done enough and we can make assertions about the data. It is impractical to create a universal checklist for the validation of a biomarker assay. I think we've all agreed to that. But a framework can be derived to guide the development of assays for biomarker qualification. This is what we have taken the first stab at in the white paper. And lastly, although the CLSI documents provide approaches to solving many of the issues associated with assay development and characterization for biomarker assays, they need to be appropriately adapted for use in the biomarker qualification space. They are not designed for the biomarker qualification space. They do, however, contain a great deal of information that can be very helpful to someone who is looking for an answer to an analytical biomarker problem or a validation problem. Comments?
Other panelists, all clear enough on these? Yeah.
I do have
Yes, please go ahead.
comment, but not on this slide, but in following up with this phase 2/3 essay that you mentioned. I wonder maybe that's possibly the confusion because biomarkers, when it comes to us for qualification, it could be just for proof of concept. Sure. So it's not a phase 2/3 thing. And using the term phase 2/3 make people think confirmatory, but we are really talking about what particular context of use is this biomarker proposed for.
I would, if we've learned nothing in the last 2 days, we've learned that there are no absolutes, okay? Everything is shades of grey. Point
And
taken.
it all depends on the context of use.
Correct.
Touche.
Okay, who's up for this next one? John
I
Michael?
can do it.
Sorry,
I mean,
Steve.
you know,
Do you want to go ahead with a comment first? Yeah.
I just wanted to offer an edit, not a comment.
We've learned that
Thanks.
is a good takeaway.
You were paying
too.
attention.
Solving seems a bit concrete in an area of gray. Addressing, maybe. And
Fair
the other thing maybe we need to highlight at the current moment is they're currently not universally available. So
They
we
are
can appropriately
they are
adapt them
for
if
money.
if
We are going to work on that. We realize that they are purchasable documents and not necessarily freely available, but they are
freely available. Perfect. Even better edit.
No, they're not yet, but we're passing the hat, right?
Yes, yes.
Actually, no, we
That's
do.
why we're doing this act up here.
That's
We
right.
expect money at the end.
So actually, was it you that accepted the responsibility to reach out and see if we can
Yes,
do something
yes,
about this? So
yes.
we point taken.
Does anyone else have any edits to this slide or the previous? Okay.
You know, after the final discussion yesterday, I think what we saw was that there's a different expectation that is around the new path to qualification, and that a final validation of your bioanalytical method is really going to be needed for the qualification plan. And so that means that we're going to have to come with a bit more data right? An understanding of the biomarker before we begin qualification, thus regulatory ready.
Just wanted to add that we will prepare you for that. In the letter of intent step itself, we'll ask you if your assay is analytically validated, and we'll also ask you if you have a statistician on your team.
Among a host of other questions.
Yeah.
Yes, sir.
Absolutely.
You know, I like your comments, your opening statement yesterday that you said, you know, you're an action-driven person, and I love the way, you know, okay, we got to go ahead. What is the next step, right? So if I may ask, what is the next step for FDA in terms of what is the timeline to get a guidance out?
Excellent question. So if you look at our PDUFA 6 commitment letter pending PDUFA 6 reauthorization, I just reviewed it right before this session. And it says that before FY 2020, I believe, and I'm looking at others in the audience who can confirm, but I'm pretty sure in the commitment letter, it says before FY 2020, we are issuing draft guidances in this evidentiary consideration space. So that gives you a target date right there. If PDUFA 6 is reauthorized in FY 2020, that means it's October 1st of 2019. Okay. And what are we? What's today? 15th of June, 2017, right? So it's saying right there that put some parameters around trying to get some concreteness. Does that help? It does. Okay.
So I have a quick enhancing edit to that first bullet point. We talk about full validation. I know there is a space in the document where we refer to partial and all of that, but it's better to be very explicit to say that the validation and all those 7 core elements identified in the paper. That's what we mean by a full validation. So it's not subject to interpretation. Somebody could come and tell you, I've done these 3 parameters fully, right? That's not what you mean.
So we should substitute validation of key parameters as outlined in the document.
Or around that, because as we've all said, the context of use drives the level of, so we could say the appropriate validation based on the context of use.
Sure, I just hope we can have that conversation to identify exactly what that is before, you know, we do the qualification plan, right?
We heard that loud and clear. We got it right. Yes.
Right. And also I think it's important not to say that some of these parameters are optional. What we are saying is the acceptance criteria for them may be variable depending on the context of use, but you have to have a strong justification if you're not planning to demonstrate selectivity or specificity. So either way, you have to address it.
Right. Thank you.
Good point.
I think this last discussion has illustrated the next bullet.
Can I just make a comment?
Oh, yeah, please.
Sure.
And just again, if it wasn't perfectly clear last time in the last session, what will make certainly the editing, is that the right word we're using right now? Is that the new hotness? Yes. Editing. What will make our editing process easier is not strike through line by line, copy chunks, provide content with appropriate reference details as direct replacement insertion in a full manner. Not a word here and a word there and a word there, because we can only imagine a fraction of the pain that John went through. And quite frankly, I don't want to have to deal with that. So please, we ask that you make our life a touch easier by sectional rewrite with appropriate references as we contemplate all of the feedback, if you'd be so gracious.
Can I ask the writing group a question, because I heard 2 different schools of thought. One school of thought said, give us the Word document so that we can change it. I heard another school of thought, which was, give us the PDF with line numbers. You're the working group. Tell us how you want to disseminate it.
So we have to date within the writing group doing it all with track changes in Word. And that works well with a limited number of people. With a much larger document, it is probably a better idea to go out as PDF'd line numbers and then have people respond with line number X to Y. This is what it says, this is what it should say or should not say. And that would be a much easier way to deal with hundreds, hopefully not thousands, of comments.
Okay, that's helpful.
Is that acceptable to everybody? I think that's manageable from our side.
And actually, from the writing group standpoint, if we could have a couple of days to actually get this out in the format that it needs to be on the website, that would be very helpful. So
Right.
early next week.
Okay, and with clear distinction of how you expect to get
Yes.
comments. And also, what did we say the deadline was? I think Martha said the end of August?
August.
August for commentary. Did you want to do the big reveal?
Go ahead.
No, you do it, mate.
So actually, we actually would, in our section, was going to have comments open for a period longer than August. But it doesn't really matter if we think we can get the comments faster. If people need to socialize this to their professional groups, it may take longer than an August 8th date to do this.
Let's ask the folks here. What do they think? Is August too ambitious?
So this is what happens when we had two groups deciding on the same thing in non-communication with each other. We got two different dates. Surprise.
Are there objections to the end of August? Okay. Okay, we're hearing an early fall, and we're approaching the month.
End of September?
End of September?
So I would just offer that and for those of you, because some of you were involved in the workshop, the framework discussion, it will take longer than you think, and you will get most of your comments in the first 2 or 3 weeks. And so, you need to understand what you want to do with those comments before you go to pharma, LD Kit, or the medical council, or whatever, because they're going to want to see something that's mostly done. So personally, Joe's suggestion would be go early, get it to something that's almost final by the end of the summer or something, then go to your bigger associations like pharma and others, and get kind of say, okay, this is like the 95% thing. Will it cause an uproar, and then work from there? I think that's just and then you'll end up having it before the end of the year. Right.
So did I hear the August 8th date?
I heard end of August. What did we
End
hear?
of August?
We heard end of August. Mhm.
Okay. Well, let's keep it earlier rather than later in that point.
Okay.
That's still 2 full months and 2 weeks on the Jersey Shore.
Okay.
So,
Okay. Anything else on this slide? Okay. But we may not be able to advance. There we go. Okay. We took the first bullet, right?
So we've sort of thrown out a putative execution date for a cleaner version, which was the end of year. Now, you just made us do a reset. We thought we'd be closer to finishing, but yeah, okay, that's a bucket of cold water, but a really harsh dose of reality, I guess. So our goal is comments and then a clean version circulated certainly well before the end of the year, right? So one set of feedback, recirculate, and then end of the year make a decision if there's 95.5, whether we go, whether we don't. And I guess it's an open question then. Is it follow-up? Is it more workshops? I guess that's the open question. What do people want?
well, I think it's where it is at that time. So we may or may not need another workshop. We may or may not need a teleconference with appropriate people. We may be able to go directly from there.
Maybe, Steve, again, it's a continuous process, like a validation, right? So you got to get the feedback from everybody. What if, again, we don't need to set up another consortium, but I like the idea that was just proposed. But on top of it, to get one more final feedback from people, right? Mm-hmm. And hopefully it's not 2 steps forward, 3 steps back. Then hopefully it's more confined. We can take advantage of these other AAPS, the NBCWRB, the other meetings that are coming up, to at least solicit one more round of discussion, hopefully, with a similar, open up the WebEx and all that to get some more feedback. One final round to either say what you want to say or that's it. Let's move forward.
So we've contacted a lot of those groups already. We've made overtures to 80% of the analytical societies, professional societies that were talked about today. So there has been some contact with them, and they have all been encouraged. And as far as I know, they are all participating in this meeting either here or online. So they should be available to provide comments in a more rapid fashion without going to the meetings. As you well know, we did that at the AAPS NBC, WRIB in the last couple of months as well. What I meant is that after this
Round of discussion, right?
It
After
is,
that
but-
one, we do one more quick, hey, here's a
But-
cleanup version.
As you said, your own clients would not permit an iterative validation to go on forever. We need to set a deadline at some point and deliver the goods.
Touche.
I agree with that 100%. We have to set that deadline and deliver.
So we're loosely considering the end of
Okay.
End of August.
End of August for
and
comments,
then
end of the year.
End of the
a
year
date
for-
before the end of the year as a 95% completed document.
Perfect.
That is our goal. I just wanted to add, I think perhaps, Shabri, you're going to execute the last bullet, are you? But I want to say that, again, as we talked about this being one of a body of documents that will perhaps engage and support Big G guidance, I want you to remember, I am in community, me is not, but unity is in community. And I ask all of you, we all have to live with this. We all have to live with the... I thought that was good, Russ.
Flying monkeys.
I had your flying monkeys.
That's the second time.
So I think it's important that we take advantage of a change. We've had wonderful documents that have served us. BMV 2001, the Lee papers, many of those 2006, 2009, the white papers from WRIB. We have had a change in context, change in language subtly here, because I think it's a 21st century problem, and there are some other aspects of clinical medicine that are following through from this. So this is a bit of a chance to take a different step down a different path, but it needs to be done in a manner that's not painful for everybody. So I really look forward to allowing Steve to do all the hard work and edit all of the comments, and I get all the glory.
I second that.
Sounds good to me.
Okay, any other comments on the publication piece, white paper? I think we've articulated that, and I've already articulated at the beginning, and with your ask, I cannot emphasize enough the analytical validation issues we all know are building blocks to anything else we want to do. I enjoyed Lisa McShane's comments at the last panel where she said this impacts ethical use of samples and ethical use of patients in trials. And so we've got to get this right. And I think with these important piece of legislation with 21st Century Cures, it gives us an opportunity to shine a bright light on something that we all know, not only for the academic community, but for industry and for consortia, this is a critical piece. So I appreciate everyone's time in helping us get this right.
Other comments from the panel, themes, ideas that we haven't already covered as next steps?
One thing that I heard loud and clear was more examples are needed. And we'll have to come up with hypothetical ones because we are limited by the clinical biomarkers that are qualified. And we could probably take one of them, but would people be comfortable with hypothetical examples, or does anybody want to contribute their case studies as examples? So that's something else. I mean, you could hide the name of the biomarker.
I'll offer a comment I saw at something very rare. I think happened at a stroke meeting, the STARE group, a few years ago, which was a company. So FDA usually, we can't reveal any confidential information. So you want to go like, hey, tell us about the failures. They're really educational. And we go, we can't talk about that. But if a very noble company comes forth and talks about a failure, then we can comment on it. And it takes a lot of guts to do that, a lot. So I don't underestimate that. But in terms of examples, there is nothing like that for education. So I just put that forth as if someone takes up that challenge.
Thank you.
Yeah, it's a great point, and I think that there will be some people that do have case studies, real examples of both positive and negative outcomes, and I think they both have a part to play. Certainly, before and since the Lee paper, some of us have been given educational workshops along those lines for quite a long time. I certainly have case studies I'd be very willing to share of real data. One of the things I would encourage the community in general to do is when we're working as service providers or pharma companies, is not to be afraid to share data. Just to give you a sort of an idea of how bad it can get, about 2006, I revealed and reported at AAPS of a commercial assay that was then being used on practically every Alzheimer's study in the world that was totally inappropriate to measure A-beta-42. And within 2 weeks, I had several contacts from certain companies developing drugs, all admitting that they knew there were issues, and they never told anybody about it. So that's the bad side of it. When I do get really interesting data, I always ask our clients if we can share that, even in an anonymous way. If it's practical data that demonstrates how these experiments tell us what how to characterize the method, then it should be out there in the community. We're all here, hopefully, to help eventually benefit patient care and welfare.
Can I just respond to that briefly? So I want to just add a point. So Clinical Chemistry, the journal, has very, very, very many examples of the example we showed, the method comparison studies, a lot of inter-instrument, inter-platform method comparison studies do and have always been published in Clinical Chemistry, the journal. And that is actually a wonderful journal, and perhaps a friend in this particular setting. Many of the markers we've described today across many platforms with 6-folds in slopes, even though they are harmonized to get the same number from a reference standard, you get 6-fold changes in slopes when they measure real people. So I would recommend Clinical Chemistry. Thank you for bringing that to our attention, John.
So go ahead, John. Did you want to respond?
I was just going to say, you and I come from the same background, so I know where you're coming from.
Okay, you guys can have a beer together later. So, but I want to mention exactly what John said is where we need your help. Okay, I'm sitting on hundreds and hundreds and hundreds of case studies. Okay, there are companies that are looking for the same biomarker for several different indications. That boy, some have worked, some have not worked clinically. My request is that I think the exact same thing John asked is that when we come to you, the pharma, and if we can go in and get permission from you to share that and put some of that information in the paper in an anonymous fashion and make a biomarker whatever to biomarker ABC, there's tremendous amount of data we have. But at least we met some of you guys here. We know we work with your colleagues in the pharma. So we're going to come to you and ask permission from you to go work with your colleagues and with your legal so we can release some of this. I mean, it would be very helpful. So John, I agree with you.
So I had one other question actually to posit to all of you. I'm actually very encouraged that we're talking about augmentation and not tear it down and burn down the house and rebuild. So augmentation is great. But how big do you want this document? Are you worried about the size? There were some comments yesterday. It's a 50-page document. If we augment everything, we'll be talking about chapters, not a document. So do we have to worry about the size of this thing? Really? Please provide
So a good question to follow up to that is why? If the document is not complete, it's not going to be of use. If it is 80 pages long and it fits the bill perfectly, then it's the right length.
And the examples can be added at the back as appendicites, whatever.
Yeah, but I mean, they count as pages to a document. So this should not also be the Cliff Notes guide to biomarker assay validation with a bunch of appendices for you to figure out. But it should be the scientific guidance on how, little g guidance, scientific advice on how to do this properly.
I don't disagree, and certainly the working group will have to figure out what they're comfortable with. I will say on our side, we have a history of creating long guidance. And
white papers.
No. And we're being encouraged to go forward with short guidances that actually focus on the key points so that people can take that and actually use it rather than try to find it buried within the 50 pages. So to the extent that you guys can work together to create a 1 or 2 page of bullets of the key take-homes, that might be a way to have your cake and eat it too.
So let me rephrase the question then in light of his answer. should this be complete with the information that is needed to do that? So No. But brief is an arbitrary term. Should this be comprehensive enough to allow someone to fully understand how to validate a biomarker assay for biomarker qualification? Yes or no? Yes. Then if we have that complete, the document is the right length, whether or not it's 5 pages or 100 pages. We already know that 5 isn't going to do it. I personally hope we don't get to 100. But if it may take that with all the examples, then shouldn't we do that? And if it's well written enough, we can go to the point in the document that addresses the point we need help with, look it up in the index, and there it should be.
So can you generate a summary that you publish independently so that people actually know that your document, your 80-page document, has the information they need? Because many labs, when they download your 80-page document, are going to say, no, there is no way I can find what I need in this document. However, if they read a summary and say, oh, look, in this document is something that I need, they're far more likely to go look at it.
So to recapitulate, we could certainly provide an executive summary of the document in brief of really what the document should cover, and then let people go to the detail.
Can I just change one word? It's our document.
Yes, thank you.
Please.
Yeah. So I will
Microphone.
It will be a peer-reviewed paper. Yes.
I've been trying to put my head around this for the last 2 days, trying to figure out what this paper actually is targeting. And I kind of, we've talked about that there's 90 or 95% of the work from biomarkers don't go to in vitro test. They don't end up being on a clinical analyzer. They're used for other decisions. So it's sort of like saying people, we have 100 people and we want to send them to Washington. We say, but 5 of you are going to go to Antarctica. Everybody put on your snowshoes and let's go. I mean, it's in this time of year too. I'm a little concerned that this is being driven for the extreme as far as the papers being 80 pages long, when we want to be sure that we get the 95% to the right place.
So let's be careful of our 95%. This paper is targeted at one specific thing: qualification of biomarker assays for validation of biomarker assays for biomarker qualification. That's it. Anything else doesn't have to fall in. So we want to get nearly complete of that particular aspect, period. Anything prior to that or subsequent to are out of scope.
But what I'm concerned is what I'm hearing from FDA representatives here is they're really leaning toward that end of the spectrum for putting together in a big G guidance. And I'm afraid we're going to leave 95% of the people how they're using out of the picture.
But I hear what you're saying, but remember, if we're looking at 21st Century Cures Act and the PDUFA 6 reauthorization goals, those specifically speak to biomarker qualification. So yes, I think the principles that are articulated, it would be wonderful if once these principles are articulated, they could be extracted to be used by the academic community in a much more understandable way for that group or for other communities at NIH or what have you. But just to be clear, kind of where we're headed with all of this is really just that, like you said, that perhaps 5 to 10%, that's really about biomarker qualification specifically, right? And what level of credibility and reliability of assays we need for just that piece, right? So, but I think the principles are valuable, but what's going to inform perhaps future guidance is really going to be that small subset. Does that help clarify?
I think so, but also
I'm really curious what your comment is here, because you get our stuff.
Yeah, so take the 2001 guidance. That was a PK guidance. Then the 2013 revised draft, it was still PK assays. And remember those discussions we had at these bioanalytical, forums where you had the chromatograph group there and the LBA group there, and they were, arguing, well, ours is harder and ours is more difficult. There was this stereotype that PK assays, that was more straightforward. How did a 2001 guidance then become almost double the size in the revised? Because there were more scenarios that had to be captured as a result of publications that happened over a span of 12 years. The reason I'm bringing this up is because regardless of the size of what we talk about the big G guidance, regardless of the size of that, there are way too many stakeholders involved in biomarker assays. I don't care if it's academia, CROs, or for other research purposes, that, and I think you're alluding to this, that may go to this document and do a control F to find, well, what do they say when it comes to bias or precision? So there seems to be, and I don't think anybody here explicitly said this, but I've heard it at the water cooler conversations or wherever else, people are yearning for this all-encompassing document. However, that's a difficult one to write up. Now, given what I said about the PK example, imagine all the scenarios for biomarker assays. A lot of you that are familiar with generic drugs, you heard of product-specific guidances. So for this particular drug, there are specific guidances that will talk about in vitro tests that you should do. Arguably, you could actually come up with, and I am not suggesting this, but what I'm saying is biomarker-specific guidances, because each one may have their own characteristics when it comes to precision, the technology used. The challenge here is just what we saw with the PK realm. There are more scenarios, and it's very difficult to predict how robust this, if there is a big G guidance that is a result of all this, how that'll look. But what I'm trying to emphasize is that there are a lot of stakeholders that will do a specific search to see, well, I just need to know how to address stability. How many samples? Some people are talking about that we do need to have specific approaches. The reason is because of the lab-to-lab variability in the approach, which is why somebody from Merck yesterday was talking, Frank from Merck yesterday, I think he was talking about, what about the number of samples? What about the number of repeats? What about the number of days across the days? How many? That is something we have to really think about if we really need to go down those specifics. Some people say don't want to be prescriptive, but please understand why sometimes that is the outcome, because they just want a baseline. Where do we start?
Sorry, just one comment to this. I mean, I hear a lot of concerns about it, and we hear that we have a lot of different stakeholders here. And to be honest, I think the first version will not be 100% perfect, but why not looking at the good side of it? I mean, just get it finished fast, put it out there, Steve? And then maybe meet like 1 year, 2 years later, whatever, and discuss how it was working, and then maybe refine it then, get comments on, okay, so this didn't fit really, and this was a pain to do it, whatever. But I think, we only will know when it will go live, what really is appreciated and what may we need to refine, and not having all concerns, because we will not be able to have all concerns fit in that document. That's just the comment.
Well, to paraphrase, better is the enemy of good enough. And of course, good enough is an arbitrary term like 46X.
Did you have a follow-up?
Thank you. Go ahead.
I just want to say that I agree with what John said. To have a document that is totally prescriptive of all of the scenarios, I think I would be very frightened if I was you. Think about all of the different biomarkers, the combination. We have protein, peptide, straight mass spec, digestion with mass spec, capture, digest mass spec, immunoassay, and this is all singleplex. What happens when now you want to take it to the next step of having multiplex assays? I think it's nearly impossible to have something that covers all scenarios, and if you did, it would be a 1,000-page big G guidance.
So I think if I want to so one thing you just said, which we may have, sorry Frank, you're not in the room, we may have put a stake in that one, is multiplex. I hope you can appreciate the dimensionality in the conversation, just talking about two technologies and a singleplex right now. So I hope you'll give us a bit of a pass on multiplexing in this one. Not saying that those concepts cannot be translated from whatever comes out of this, but that's not a problem doubled. That's a problem powered.
Yeah, agreed.
Right. Any other comments? Yes. Or edits, I'm sorry.
It's a comment. If you don't address exploratory biomarkers, but you also want to address the academic community, be aware you have to tell them, even for exploratory biomarkers, we have to have technical validated assays. So I think it's also a type of education that this assay validation is not only for the biomarker qualification program.
So you make a great point, and there are a number of authors here. I'm looking at Russ. I don't see where Jean is, but John, can we refresh the Lee paper and just translate some of the CVI, CVG? Because I think that framework fits that problem, but we have a disharmony in language. Can we think about that as an update mechanism feeding into this and then whatever comes from it? Is that possible?
And the other comment, I wouldn't mind if there are dozens of examples added and the document is getting several hundred pages, because let's say examples of whatever type of biomarker qualification helps to understand at least the academics much easier, and you don't have to read all those. You can pick.
It's a fair point. How are we going to keep our wives happy when we're writing all this stuff for mate? Appreciated. If you have any specific feedback on the flavoring, so we've talked about safety biomarkers, but we haven't talked about different functional biomarkers or use of. Again, if there's some specific examples really rigorously listed in literature, that helps us deconvolve and translate. And the more crystal in the examples, the better the quality of the data. The more open those examples, the better we can convert that into a story, really, if you want to think about it.
And maybe you also can add, let's say, examples, the links to our letters of support material, which we have submitted to the FDA or EMA. That could also be a possibility to have a list of those links added in the appendix. Otherwise, it's great. Bring it on fast.
Can we stop now?
Good. Thank you.
Thanks. I think maybe one or two more comments.
Russ, I can't speak for Jean, so I would leave Jean as the lead author to respond to that. But I think what would be good is if we can keep the text of this document as brief as possible, but I would encourage more case project studies, real examples in an appendix, and preferably as many as possible categorized by those 7 core components. That if you get this right or wrong, this is the potential outcome in each of those 7 categories. I think that would be helpful for people to address some of the comments that have been made.
Yeah, I don't doubt we're going to get that in a clear, concise manner from you, John, so I appreciate that. Thank you.
Any others? Okay, Shavri, any final comments?
No,
thoughts?
just a big thank you to everyone who stayed till the bitter end. This is really, really helpful. I know this is a lot of work, but we're committed to helping move this forward.
Yeah, and I too really want to thank the writing group, the presenters, the panelists, the commenters, or I'm sorry, editors, who all contributed to the discussion here, and what sounds like will be some valuable refinements to a very important white paper. This has been a lot of time and effort on a very serious topic, and I think the interest from diverse perspectives and all the people who joined us in the room, we had hundreds who joined online too, shows just how important this issue is for this overall goal of evidentiary considerations of biomarker qualification. So it is foundational for that work. There are more steps to come, but very important progress here. I particularly want to thank John Michael Sauer, Martha Brumfeld, and Nick King at CPATH, and Shavri, of course, and all of the FDA colleagues who have put a lot of time and effort and collaboration into making this work as relevant as possible and in making this meeting a success. And a final thanks to my colleagues at Duke who have worked to help put this together with CPATH and FDA, Meredith Free, Joanna Higginson, Jessica Burnell, Catherine Frank, Grant McHorse, Elizabeth Murphy, and Liz Richardson. So thank you all for your participation. Please enjoy the rest of the afternoon and safe travels back. Thank you very much. Yeah, this is quite a significant undertaking. Thank you very much. Great. Thank you. Actually, we've had impressions before at our events. Never Yoda. Nice to meet you.